All right, hi. Welcome, everyone. I am so happy to be up here introducing today's session and appreciate everybody who arrived early in order to attend today's session. I want to start with a thank you to our sponsors. It would not be possible for us to have this event today without the really generous support that was provided by all of the groups that are listed up here. Akuja Therapeutics, the Acoustical Society of America, Sound Pharmaceuticals, Turner Scientific, CBSET, and Silcare, and additional support from the Richard Bellucci Translational Hearing Center at Creighton University. I'm really grateful to all of our sponsors. Please thank them if you have time. They're responsible for this room, this event, for the coffee break that we will have, and for the reception that we'll have tonight. So thank you to our sponsors. And thank you to ARO and to Parthenon Management Group also. I don't think they're in here right now to hear their thanks, but Kristen and Laura have fielded probably 300 emails from me in the last 2 weeks, so I'm really grateful to them. So welcome to all of you. This is a satellite symposium that we're doing, really for the first time at ARO. The history of this meeting, 10 years ago, there were several symposia that were held in conjunction with the Office of Naval Research, and we have been really eager to bring that back and to bring it to a larger audience. So I'm really pleased that the Hearing Center of Excellence was supportive of renewing that seminar series. The Pharmaceutical Interventions for Hearing Loss Working Group, if you're not familiar with it, has put together a number of guidance documents over the past 10 years, probably many of which you've seen in the past. Within the Hearing Center for Excellence, Commander Amanda Boudreau, Dr. Teresa Schultz, Amy Buden-George, Danielle Lester have provided a tremendous amount of support. If you have ever joined one of the PIHL quarterly calls, it is thanks to the great support at the PIHL that we have those phone calls. They were instrumental in getting the government approvals for us to be able to propose and hold this meeting. If you would like to be on their mailing list, I have a QR code coming up, and I'll share that with you so you can get your phone ready for that. In the meantime, I want to welcome two HCE representatives. We have Dr. Quentin Hecht and Ms. Amandria Berzuska here with us. I'm going to ask them to stand and wave so you can see who they are. They are happy to field your questions. If you have questions about how to interact with the HCE, who's at risk within the military, what are some of the major hearing challenges, how can you address research questions and partnerships with the HCE, these are your contacts, so please find them at coffee break. Please use them to get information that will be helpful for you in your planning, and please thank them for being here. I mentioned that there are a number of guidance documents that have come out of the PIHL working group in the past. These are all open access. You can see that we've covered a lot of ground over the last 10 years. The earliest documents were very focused on the current state of the science for pharmaceutical interventions. We have had special issues focused on noise in the military, on cellular mechanisms of ototoxicity, on clinical monitoring, noise-induced hearing loss translating risk from animal models to real-world environments, covers everything you ever wanted to know about mice, guinea pigs, rats, chinchillas, monkeys. Many of the people in this room right now have contributed to a number of these documents. You'll also learn everything you ever wanted to know about octave band noise, impulse noise, pure tone exposures, blast exposures. So I encourage you to take a look at those papers. The most recent collection was also in the Journal of the Acoustic Society of America, and while it was focused on noise-induced hearing disorders, it was really intended to start facilitating conversations about standardizing some of the test measures that we use in humans. There's papers on the audiogram, on otoacoustic emissions, on electrocochleography, on the middle ear reflex, everything you might want to do within a research study or a clinical trial. We have a document summarizing how those measures have historically been used in research and in clinical trials. So I invite you to take a look at those, and I invite you to help contribute to upcoming documents that we hope to continue to prepare over the coming months. Some of the current and upcoming activities for this Pharmaceutical Interventions for Hearing Loss group, we have quarterly meetings. They're currently the first Tuesday of the month. They have been at 3 o'clock Central Time, but we are in the process of moving those to be in the morning in the United States and in the afternoon for our European colleagues. We have a great international presence within the PIHL working group, and we are trying to make the meeting times more convenient for the entire group. This is an open group. We have people who belong to academic institutions. We have people from industry. We have members from foundations. We have members from government. Really, all stakeholders are invited. So if you're not familiar with the group and you're interested in it, you have an open invitation, and we are a volunteer community. So we are always looking for people who are willing to help generate those next work products. Some of those next work products, we're planning reviews of animal models with the goal of possible standardization or recommendations for standardization. We plan to review existing human clinical trial designs. Again, for possible standardization, recommendations for standardization of test measures, and also to talk about lessons learned. There's been a huge amount of research in the past 10 years, and there's been a lot of obstacles encountered along the way, and we'd like to put together information that will facilitate everybody's progress. If you'd like to join the PIHL mailing list, I have a QR code for you. I'll pause here for a moment. If you don't grab the QR code, you are welcome to reach out to me. I'm easy to find, colleen.laprelle@utdallas.edu. Our schedule of events for today, we have a lineup of speakers across the scope of basic translational and clinical research, industry services, clinical services. We really want to use the PIHL session as an opportunity to help everybody advance their programs, whether you're somebody working with animal models who's interested in how do you get to the next step, whether you're working with human patients already or human participants already, and you're interested in developing your next study, whether you're interested in understanding who your possible collaborative partners are. We're hoping that you'll leave here with something that will help you advance your program. We'll have our first 3 talks from Jonas Deerfield-Johnson, Gail Naert, and Ralph Holm, and then we'll have a question-answer session. We're hoping that you'll have questions that help us have a productive discussion. We'll have a coffee break, and then we'll have Drs. Amanda Henton, Tim Balke, Jonathan Keel, another question-answer session, and then we invite you to join us in the Atrium C. We have a hosted reception, again, thanks to our generous sponsors. Akuja Therapeutics and Sound Pharmaceuticals are sponsoring beverages and some light snacks, so please come join us for the reception at the end. I'll put this slide back up. You can't give us feedback until we've actually shared something with you. So I'll come back to this later, but we are hoping you'll provide feedback. We are interested in making this a repeat event, and so we invite your feedback on whether today is useful and what topics you would like to see and who you'd like to hear from if we are able to offer this event again in the future. With that, I'm going to stop and I'm going to introduce our first speaker. It's Dr. Jonas Deerfield-Johnson. He comes to us from Akuja Therapeutics, where he's the Chief Development Officer and a Managing Director. His title of his session is Drug Development 101, an Inner Ear Scientist Illustrated Primer. Please help me welcome Dr. Deerfield-Johnson.
Thank you, Colleen. first of all, I think there's one person that hasn't been thanked yet, and that is Colleen herself, because without her efforts throughout the last year, this symposium definitely would not have happened. It's to some extent a lot easier to donate some money to sponsor something than it is to pull together everything and do all the organization. So please give Colleen an extra hand for this. All right, so giving a sort of a broader introduction talk, I think the first question is always, how do we come up with a title? And I've always been a big fan of science fiction, cyberpunk as a genre, and I think it's interesting to know that we're now living in a time where many of the predictions from this type of literature have come through. We have personalized medicine, gene therapy, lifespan, health span extension, etc., a number of technical advances too, although we're still lagging behind a little bit on teleportation and space travel. But one of my favorite authors is Neil Stephenson, and he wrote this book, The Diamond Age, or A Young Lady's Illustrated Primer, some 30 years ago. And in this book, a young poor girl receives a stolen copy of a particular device called a nanotech primer, which is a children's or young child's educational adaptive device that's supposed to meet the kids where they are and bring them forward towards being members of society that can disrupt the status quo and help bring society forward. So that's where my inspiration for the title of the talk came from. I can highly recommend the author. If you want a few recommendations from him, there's a couple of them listed here. I think, interestingly, the last book he came out with in 21 explored geoengineering by shooting sulfur into the atmosphere to reflect sunlight and combat global warming, and that was actually just reported as an unintended consequence of cleaning up shipping pollution over the last few weeks, that less pollution from international shipping with tankers had reduced the amount of sulfur in the atmosphere and potentially unintentionally contributed to global warming. In any case, keep reading good literature. It's better than doom scrolling and hanging out on social networks. So when it comes to drug development or pharmaceutical development, I think many people come in with sort of a black box view. You have a great scientific discovery, and then you run a clinical trial, and then perhaps for the more cynical ones, then you get rich. But in reality, it's a bit more complicated. And this was a great review that was published 8 years ago in Nature Reviews of Drug Discovery that really maps out all the intricate steps that needs to be taken to have a reasonable chance of actually eventually bringing an idea out to patients and to helping especially those who are in underserved medical areas. So this is a very simple topic for a 45-minute talk. I won't get into everything. I won't hit all the details, but I see a lot of people in the room who's also been there, done that, so I'm sure the reception will give plenty of options to discuss also with them afterwards. I will mostly take the perspective from small molecule drug development. Still constitutes a lot of the new drug approvals that are coming out, but there's very similar steps and processes to take if you're dealing with cell therapy or gene therapy or any kind of other modality that you could envision here. General principle says that the later you get into development, the more expensive it gets. So it's a good idea to de-risk, especially financially, your development program by having some critical parameters to potentially eliminate a candidate with earlier on in the development program, to not sort of run into trouble with the translation or with the ability to progress your program. Contrary to others, I would not recommend fail fast, but I would recommend being able to pivot fast. So backups or alternate ways of delivering your drug or something like that when something unexpected comes up, because something unexpected will come up, you should be able to find an alternative way forward. So if we start with the preclinical aspects, it comes broadly speaking in three big categories. You have the bucket of biology, of manufacturing, and then, of course, of some regulatory interactions. If we start with the biology, that's probably where those who are coming straight from science are most at home. This is where you will eventually start by generating your idea. This could happen in a number of ways. It can come from your original research. It can come from clinical biomarkers or observations in patient data. You could even get it from the literature or from looking at competitors in the field. What are they doing? What do you believe you could be doing better? Is a different modality the way to go? And once you have your idea, as part of that idea, you'll have your target, but you should initially, of course, want to validate that target. Does it actually have the kind of impact, the effect that you want it to have? So target validation is essentially a matter of target perturbation. What happens when you overexpress it? What happens when you knock it out? What happens when you hit it with siRNA or a tool compound? Does it actually seem to be doing what you expect it to be doing, and with an effect magnitude that you also could believe would actually be relevant for the particular disease? One thing I would want to introduce from the industry side of things is the idea of a target product profile. This is not a definite profile that you set up at the very beginning. It's sort of a living profile or a living vision of where you want to go. But the idea is that it helps you take decisions as you go along the development path. So you want to define who you're going to treat, what patient population, what pathology, how do you want to treat this patient population through your target. Is it gene therapy? Is it small molecules? Is it something else? How are you going to treat? Is it going to be a daily treatment? Is it a one-off? How are you going to administer the drug? And what kind of effects would you think would be, side effects would you think would be acceptable for this population? As an example, cancer drugs have very significant side effects. That's well known. It's accepted because they tendentially save people's lives. But you might not be accepting the same kind of side effect profile for a skin cream. So these are things you should think about up front, because once you run into these various complexities along the way, those are the things that will help you make a decision about whether you're going in the right direction or not. And then, of course, once you've gone through all of this, you need to have either from off the shelf or existing techniques, assays for screening, or you need to develop them. Now is the time when you actually want to start generating candidates for hitting this target in the way that you're intending to do going forward. And this is typically something that happens in vitro based on potency, very simple readouts. You want to be able to, at minimum, do medium throughput screening, potentially high throughput screening, depends on what modality you're working with. Is it hundreds of thousands of compounds in a library, or is it a more limited number of vectors and capsids you want to be running through? Typically, you'll get your early hits. You'll get an idea about what could work, what couldn't work. You will make refinement for small molecules. That's where your medicinal chemists come in. They'll modify the chemistry of the compounds. They look at how different side groups can affect the ability to hit your target and the potency of this. So it's more of an iterative process at this early point. You also start looking at different experience-based criteria for whether or not a hit has a likelihood of becoming a drug. What is the molecular size? What is the solubility, lipophilicity, permeation through assays like CACO2 that are supposed to simulate oral absorption if it's a drug you expect to be delivering through the oral route. Stability in microsomes, so liver microsomes. Is this something that could be predicted to have a half-life that would be relevant for becoming a drug, or is it eliminated or broken down immediately? Transporter interactions, similar things. All of them of different level of importance depending on your target product profile. Are you developing an IV, a transtympanic, or an oral drug, for example? You will do some early selectivity. It's not always given that because it hits the target that you've expressed in your screening assays that it only hits that target. This is sort of typically more in the adjacent target. So if it's a receptor family, you might want to look at how it hits other receptors in that family. And then, of course, some early liability assessments as well, looking at the perspective of side effects and what you think is acceptable. Typically, you can look at things like HERC, potassium channels expressed in the heart that are good indicators of whether a drug would cause cardiovascular problems. Inhibition of CYP405 enzymes in the liver, is it something that could interfere with metabolism of other drugs or metabolism of anything in general in the body? Or cytotoxicity, particular liver toxicity, is also something that's typically looked at early on to eliminate candidates that at this point already are unlikely to be taken further forward to the end. And then my personal recommendation for anything in the inner ear field. There's always been a lot of discussions about why we are special and why it's so hard to get something to the ear. I'm not sure that we are that special. I think a lot of the similar considerations happen for CNS drugs, and we do treat CNS disease with various modes of delivery. But this is a good time to already get an early view on whether your hits are actually likely to get it to the target, whether that's from local, semi-local administration or for systemic administration. At this point, you also still have your medicinal chemists involved, so maybe you can improve that if you have an indication of what is helping a certain type or certain class of molecules to actually reach the inner ear. So based on all of this, you will have selected a lead, a candidate that you want to take forward. Again, I would rather say leads. It's a good idea to have a couple of backups here. You're still not at the end or even close to getting into clinic, so having something you can pivot to is not a bad idea. And now you get into more either native tissue in vitro or organoids or something like that that would allow you to continue in vitro work or eventually in vivo pharmacology in disease models or mechanistic models that simulate the disease, often in our field. And there's a lot of in vivo pharmacology to do, dose response studies, treatment window studies, treatment duration studies. You have to start getting into looking at pharmacokinetics and ADME, so absorption, distribution, metabolism, excretion studies. How do single doses behave? How do repeated doses behave? Where does the drug get to? It often doesn't just get to where you want it to. Sometimes it doesn't matter that it gets to other places, but other times you want to know about it. So you might be looking at CNS, you might be looking at other tissues, depending on the target that you're working on. You'll also need to do some in vitro metabolism studies to look at which kind of metabolites are coming when the drug is broken down. These are important for choosing especially toxicology species and seeing what's relevant for the human metabolism of a drug. Could it interact with the metabolism of other drugs by inhibiting or inducing metabolizing enzymes? And how is it binding to plasma protein in the blood for a systemic drug, or for example to brain tissue as well? How much free drug are you going to have that's available to act on your target? And then it's time to get into some preliminary toxicology and safety, so not GLP type toxicology safety yet. You'll do some safety screens, so I talked about earlier looking at adjacent targets to see how selective you are, but here you're really going broader, looking at other receptors, ion channels, enzymes, transporters. Is there a risk of your candidate here hitting something else? And this will, of course, also inform that what you will eventually be looking for in the full toxicology and safety studies. What should you be aware of? Do you need to measure any particular endpoints? You'll do some in vivo work as well, dose range finding and maximum tolerated dose in vivo studies that, again, will inform the design of the next step of the GLP toxicology studies once you want to have a robust design before you sign on the dotted line for $1 million toxicology package starting from scratch. Okay, so now all of this comes up to the big ticket items at the end here. There was a couple of years ago a lot of excitement and interest around the fact that the FDA was announcing that animal testing was no longer required as part of the Modernization Act end of 2022. But this comes with a caveat. if you want to do something else than animal testing, it's on you to justify the relevance and the capability of your alternate approaches to be predictive for human safety. So everything that's been done up to now has been done in animal studies. There's a huge historical database of which species are predictive for what is relevant for particular safety concerns. If you want to come with something else, and it might be a great idea, and it might save a lot of animals, it might save a lot of time, it might save a lot of money, you have to convince the regulatory authorities that it's predictive for patient safety. So typically, you still have to deal with the classical so-called IND-enabling GLP toxicology and safety package, which will usually look at genotoxicity in different models and assays, safety pharmacology, so CNS, respiratory, cardiovascular aspects of safety, and then general toxicology in 2 species, non-rodent, rodent. Typically on the shorter end of things, most people do on 14 to 28 days, repeat those studies to be sure that they cover a bit longer than what a typical phase 1 study would look like. Maybe also take them through an additional type of study if it's a shorter treatment duration in a phase 2A study. These have to be supported by formulation analysis. Are you really giving the animals what you believe you're giving them? And toxicokinetics, so what level of exposure are you actually getting in vivo in the animal? Sometimes you might look at phototoxicity for drugs that are systemically delivered. I'll get back to that a little bit later, what you're going to do to avoid that. And you might be doing additional work in vivo, in your in vivo models, both pharmacology, PK distribution in additional species is one thing that's quite interesting to look at, because in the end, you have to be able to predict your doses and your safety margins in order to start clinical studies. And you might want to know a little bit more about how different sizes of animals, different types of animals are behaving in terms of target exposure to have a robust prediction towards the human doses you're going to propose. So at this point, you should have pretty much everything that you want from the biology side of things. But in parallel, you'll have had to start looking at manufacturing, which is something that at least I, coming from academia 15 years ago, was not very aware of all the challenges that came in that side of things. So you'll often hear the abbreviation CMC. It comes from chemistry, manufacturing, and controls. I think everybody perfectly understands that you need to be able to do some chemistry to synthesize a molecule, but really a huge part of the work here comes down to also setting up your robust manufacturing and also controlling that manufacturing to a very large extent. So you'll be working on your active pharmaceutical ingredient, also known as the drug substance. You have to now go from a lab scale production. You need a few grams of something to run lab studies, but now you have to have a robust and scalable synthesis process. You have to also develop and validate analytics, and you need to characterize a whole bunch of things around both the API, the active pharmaceutical ingredients, such as physicochemical properties. Here, one part is the UV absorption, for example, from the absorption spectrum of a small molecule. If you don't have absorption in the UV spectrum, well, no need to do phototoxicity because you won't be putting anybody at risk there. So it serves different purposes. But one thing that people tendentially don't think about is not just about the active principle, it's really about all the impurities surrounding it. So either from the synthesis itself, leftovers from the synthesis, side products. A chemical reaction is not just the one you write out on paper that you want to have, it's also what's produced based on the conditions and the excipients around it, but also impurities from degradation. Generated either at extreme conditions or during stability, so long-term preservation or storage of the compounds. A lot of this really comes down to the same as the toxicology studies. Can you prove to the regulatory authorities that you're not putting patients at risk, not just from the API itself, but also from everything that's surrounding it from your manufacturing process. Once you've got all that in place, you want to manufacture under good manufacturing conditions, the manufacturing equivalent of GLP, good laboratory conditions, procedures for your clinical trial material. but it's not finished because you also have to deliver it somehow. So you need to work on the API and the formulation that's relevant for your desired route of administration. Again, you need analytics. To a certain extent, you can take them over from the drug substance development and work on them to be relevant for the particular formulation you're now working with. You'll have to also describe to some extent the release properties, so what happens after you administer the formulation. How does a tablet disintegrate? How does a gel formulation release? How does a subcutaneous depot formulation release over time? If you have an injectable, you need to validate that you are manufacturing it sterilely, that you're actually able to deliver something that doesn't come with any kind of bacteria or other things in it. So this has to be tested for and validated as well. And then again, the robust, scalable manufacturing process with all these controls implemented, but also how you fill vials, how you finish tablets. Are they uniform? Are they indistinguishable from each other? Are they indistinguishable from the placebo? How you're going to pack them up? How you're going to carry out your blinding? This is all something that's part of this process before you can actually deliver it to a clinical trial site. How long is it going to be stable for? Under which conditions? What shelf life can you put on your product before you ship it out to a clinical test site? And then, again, the final step of GMP manufacturing for the clinical trials. With all of these complexities in mind, it's a good idea to discuss with the regulatory authorities along the way. I would say the sweet spot is somewhere between the early toxicology before we get to GLP and when you've gotten to somewhere around the technical batches, so not your GMP manufacturing, but you have some idea about how your active principle behaves and how your formulation might behave before you go into the last steps of manufacturing. This is really the chance to have a discussion with the different regulatory authorities, get an idea about what kind of concerns they have, ideally get some sort of alignment or at least approval. It's not really approval in the sense that you get pre-approval, but at least a conditional message from the regulatory authorities that they could agree with what you're proposing to do once they've seen the data. And this is before it gets really expensive, so before GLP tox and safety, before GMP manufacturing. So you'll submit a package, present what you're trying to do, your overall program, and ask about 10 questions in different areas, typically the CMC, the non-clinical, the clinical area, looking at what drug are you trying to produce, how you're going to control that it is pure, that it's not carrying any impurities that could be complicated to deal with or cause risk to the patients. What kind of program have you carried out on the biology side? How do you plan to execute your toxin safety? And of course, what clinical population are you planning to treat? What's your dose? What's your protocol? What are your endpoints? Does all of this make together sense to the regulatory authorities? If you have exemptions, that things you don't believe that are relevant from the classical steps that you need to do, this is also the time to discuss whether this could potentially be agreed. And then you want to get a written response or a set of minutes from a discussion that helps you guide your development further on, but also functions as a reference for later submissions. So with all of this in hand, you're ready to apply for the permission to actually take your drug candidate into clinic. And I take here just the examples of US-based trials and EU-based trials. In the US, you submit to the FDA and to the institutional review boards for ethics. In Europe, it now all runs in a centralized fashion through the EMA in the new CTIS system, and they will then, depending on where you expect to carry out your trial, designate a reference member state that's involved in the trial, and the reference member state will coordinate the review and the input from all the different states that are involved in your clinical trial geographically. Otherwise, I just want to highlight a couple of things that are either similar or different. The organization of your filing is different from the two jurisdictions here. For the FDA, it's a modular organization. in the way that it's listed here with modules 1 through 5, but they essentially carry the same information as in Europe. In Europe, it's a couple of major documents, the IMPD-Q for quality, that is the CMC information, all the manufacturing, everything that's around your manufacturing and control process, and IMPD-S for science, which is all the non-clinical and clinical information. And then in both cases, you also have an IB, an investigator's brochure, used to be in Europe, for those who have experience with it before, used to be also carrying what's now in the IMPD-S document, but now the IMPD-S is the detailed document, more similar to the FDA modules, and the IB is becoming a leaner document also because it will eventually get published, so you might not want to have such a gigantic document with all your information in there that becomes publicly available. What's really different is that in the US, you have to the FDA, submit your actual original study reports. You will, of course, have to have them on the European side as well, but you don't actually submit them in Europe. So that's part of the organization, that you submit all the individual non-clinical study reports and also the clinical study reports. Another difference between the two is that in the US, it's required to submit your toxicology and safety formats, studies in a sort of standardized document format or data format called SEND. This is currently being assessed in Europe, whether that is going to be a requirement there as well. And then, of course, timelines are not the same. In the US, it's a 30-day review by the FDA from the time they start until the time finished. You should not be planning any vacations because you might get requests for information at any point, and you only typically have a few days to turn this around, clarifications, additional information, anything like that. On the other hand, the IRBs typically take somewhat longer. It's a bit less predictable. In Europe, it's now been aligned between the regulatory review and the ethics review. These are now the part 1 and part 2. They're supposed to run in parallel and run on the same timelines. But that has also somewhat extended the timelines for the regulatory review if you're running a multinational trial. If there's multiple regulatory bodies that are involved in the review without additional requests for information can be as much as 60 days. However, if you have a mononational trial, then it's typically more on the order of the same duration as an FDA review, 30 days, because then they don't need time to discuss amongst themselves. They don't need time to align on things. This is, however, generally to your benefit when they do it, because you don't get the same question 5 times in different variations. You get all synthesized into one question, so costs a bit of time, but saves you some effort. So now you should have the authorization to actually start administering your drug candidate to humans. And this is classically phase 1, 2, phase 2, phase 3. It's, however, not completely done with biology at this point. You might be looking at additional pharmacology, either because you observe something in clinic, or because you still have things to clarify for later clinical trial phases. You might have further PK and ADME to carry out. There are some studies like broad transporter studies that are typically deferred until after the early phase 2 studies. And at some point, you're going to have to carry out developmental and reproductive toxicology, carcinogenicity for bigger clinical study populations, unless you have a good reason to get an exemption for this. You also have to continue on your manufacturing. There are still things with impurities that have to be tested and documented. You have to show that you can do multiple batches. At some point, you want to have more than one supplier site to show that this is a drug that could eventually be delivered reliably to patients if it gets approved. You might also be changing your formulation throughout, because it turns out it doesn't behave exactly the way that you expected when you started out. And then, of course, you'll have continued regulatory interactions, end of phase meetings, filings for the next step, and eventually a new drug application or market authorization if you are successful throughout. So phase 1 is your classical first-in-human studies. Here, the primary objective is really safety. Overall, you need to know if all the information you gathered and the doses you proposed are actually acceptable or create any kind of problems that would bar you from testing in your patient populations. And then as a secondary objective, dose exposure relationships. Is all your dose modeling, your pharmacokinetic modeling panning out? Or do you get the level of drug exposure in humans that you expect is necessary for efficacy? So this has to be validated with blood sampling, with analysis, and ideally adjusted as you go along so that you can eventually get to a dosing that is meaningful for the later clinical stages. Typically, it's split up into two types of studies, phase 1A in healthy volunteers, often in a dedicated phase 1 unit, or and a phase 1B directly into patients. This is typically done if you have a concern about side effects or that can be related to the drug itself or to the route of administration. You can imagine that a phase 1 study in healthy volunteers for an intracochlear administration of a gene therapy is probably not that easy to get through ethics if you have no potential benefit of subjecting yourself to this. Similar to cancer drugs, very strong side effects. You don't typically give them to healthy volunteers. It's often a combination of single ascending doses and then multiple ascending dose studies. So first you step up as far as you've planned or as far as you're allowed to once you start seeing potential side effects, and you pick a dose that you would then administer for a longer duration, about a week, to see if you can see any difference in terms of behavior of the drug, in terms of exposure or side effects when you give it for longer than a single dose. And again, interim PK analysis is great to help your dose escalation. Don't do it blindly just because of the side effects, but try to get readouts throughout so that you know you're not exceeding what you wanted, but also that you're not undershooting what you wanted. And again, a recommendation is might seem weird to do, but in the phase 1, it's not such a bad idea to do it blinded and placebo-controlled, because especially phase 1 studies in a healthy volunteer unit, one person gets back pain, everybody gets back pain. One person gets headache, everybody gets headache. If you have placebo controls in there, then you can demonstrate that this is a sort of a psychological group effect and not something that's actually associated with what you are testing, and you don't get that written in the file and taken forward as a problem. Phase 2 is your first real meeting with patients. This is where the rubber meets the road for most people. The primary objective is now efficacy testing. You want to see if all your hard work and efforts has paid off and it's going to do what you expect it to do in the disease indication you've chosen. You'll, of course, continue safety testing as well. There's typically, although there's no formal distinction here, typically a breakup between phase 2A studies that are smaller proof of concept studies and phase 2B that are larger dose finding studies. This might not apply to everybody. You might not need a large multiples of hundreds of patients in the indication you've chosen, either because the indication is small or that you, for other reasons, believe that you can manage with a smaller number of patients. You typically have a longer duration of treatment and observation than you do in your phase 1 here, but of course it still has to be supported by your preclinical toxin safety package. So typically you will not be able to treat for longer than a month if you only have 1 month's toxicology studies. This is again double-blind and placebo-controlled design. What's important in a phase 2 compared to your final phase 3 studies is really the opportunity to explore a broader complement of clinical outcome measures of endpoints, because we still don't know at this point really if the drug is going to behave in humans the way that we expected. I think in our field in particular, there's also a number of limitations in terms of what you can do in animal models, what you can do in humans. We still don't have a good predictive measure for speech recognition in the animal models. So we might be picking endpoints that we believe are going to pan out in the clinical setting, but we don't know until we actually do the clinical trial. So having a number of extra secondary endpoints and even exploratory endpoints to try to find out where it is you actually see the benefit and whether you need to change your design for later phases based on what you've learned here, this is the moment to do it. This is the opportunity to learn more. And then finally, in phase 3, primary objective is to confirm your efficacy, but also to compare as a secondary objective your efficacy and side effects to a standard of care. Again, a bit complicated in the inner ear field. There's typically no approved drug as a standard of care, so you might be working with sort of a standard practice instead of a formal standard of care, which can even be more difficult because it's not the same in different countries or in different regions of the world. This is something to overcome. This is why you need your regulatory meetings. This is why you need to have pre-alignment of what's going to be acceptable and how you're going to combine that potentially in multinational or multi-regional studies. And then, of course, you broaden your population, larger groups of patients for safety testing. You might run into rare side effects. You might run into side effects that are specific to a particular patient population. This is the time when you are going to have the chance to see that, or hopefully not. You might also still be asked to carry out other potential clinical studies. This could be cardiovascular safety studies, dedicated QT, mass balance studies to look at how a drug is eliminated, special populations. If, for example, you have particularly renal elimination from your mass balance study, you might have to carry out a study in special populations with renal impairment to see if that makes a difference. And also drug-drug interaction studies, for example, driven by interactions with particular metabolizing enzymes or interactions with transporters that could be involved in other drugs that people are taking at the same time. So this is not really the safety of your drug relative to the patient, but the potential impact of your drug on another drug with an impact on the patient. In the end, if you come this far, you've done a great job, then you're ready to file for a new drug application in the US or market authorization in the European Union. And then you may do what people are not necessarily so familiar with. You might do phase 4 studies. You might want to continue doing it. You might sometimes be obliged to do them. Post-authorization surveillance, get more information about how the drug behaves in real clinical practice under less strict conditions than what you see in the case of a clinical trial, and also potentially further safety information. What I didn't emphasize so much at the beginning, but you might want to do this at an earlier stage. You can see that all of this is a pretty long process with all the clinical phases. Once you even get into clinic, there are various schemes that exist to get a faster approval through both in the US and the EU, probably also in other jurisdictions. This can be designations such as fast-track designation. This can be orphan drug designations. This can be breakthrough designations or PRIME scheme in the EU. Typically, these are all based on unmet medical need or large or incentives to develop drugs for smaller populations. So these are worth looking into. This can be as early as late preclinical. You have a great idea that is already well substantiated based on your preclinical data. You might want to go see if you can get some additional regulatory assistance throughout. Doesn't necessarily lower the bar for what you have to show, what you have to meet, but it might shorten your timelines. In some cases, you might be able to get a conditional approval based on a phase 2 study with the commitment to carry out a phase 3 study and actually get your drug to patients somewhat faster. So in summary, drug development is a pretty complex exercise. I haven't even gotten to the bottom of the majority of these things, but again, I invite you to read the review paper from 2017 for those who want to know even more. It requires many kinds of expertise, not just excellent scientists with a good original idea and good research skills, but many players along the way that are experts in either the manufacturing or the development of formulations, toxicology, regulatory. How do you present all of this in the best fashion to the regulatory authorities and get the best way forward. I think it's important to say that, of course, you all know that doing your work diligently and robustly is important for your own success, but it's also important for the field as such. We are, from a drug development perspective, in the inner ear still a relatively young field. Any kind of failures have a pretty big negative impact on the perception of the field, especially from financial stakeholders, whether that be payers who don't know if they can robustly expect that what you are delivering will be meaningful for patients and will get reimbursed eventually. That's important to get it to the patients in the end, but also at a much earlier stage to get investors that are necessary to bring this forward, to help pay for all of these expensive steps along the way, to keep them interested, to keep them convinced that it's feasible to do this also for something as strange as inner ear diseases with a very limited track record to compare to. So everybody's way forward here strengthens everybody else in the field and puts us in a good position to meaningfully help the patients in the long run. And that being said, the inner ear diseases, of course, remain a major unmet medical need. You have a great potential to make a difference for patients by working on it. So I hope at least I've contributed a little bit with this primer to equip you to be disruptive contributors to breaking down the status quo and bring a bit more than one drug all the way through to a market authorization. Thank you for your attention.
That was a tour de force. We have time for a couple of questions right now, if anyone has questions directly related to this session. We have microphones in the middle. Yeah, Rick? There's microphones set up in the aisle.
So actually, two questions. One is, given the location in the inner ear, typically monitoring for exposure and correlating dose with exposure and effects and translating preclinical is relating it to exposure in blood or having access to tissues, which is easier preclinically. So I guess the challenge for inner ear, and we may see some of this revealed later, is how do you monitor for that type of aspect? Any thoughts?
No, I think you're perfectly right. It's not the same. You don't have the luxury of directly relying on blood sampling unless you combine it to a large extent, at least for a systemically administered drug, with a good understanding of the relationships between the circulating drug and the drug actually reaching your target. I think that's why multi-species PK and distribution studies are quite important. We've seen it, for example, with the Aqua85 candidate that Tim will discuss later, that if you keep the dosing volume of the same concentration, so the exact same formulation, constant relative to the inner ear volume in species of different size, you actually see an increasing exposure duration as the species grow larger and also have a larger inner ear volume, which is of course something that gave us confidence in saying, well, humans are bigger than the species we've tested, so we expect that we get at least as long an exposure or longer than what we've seen preclinically. Seen it with other programs too, that looking at what both rodents, mice, rats, guinea pigs, cats even. There's plenty of opportunities now to do mini pig PK, to even do sheep PK. That'll give you a better idea about how this relationship between blood levels, plasma levels, and inner ear levels are connected. I think again, the same thing goes for a locally administered drug. You want to try multiple species. You want to build your data set, your understanding for making this dose translation. Also taking into account again different volumes, but also for local delivery, different thickness of round window membranes, everything that could affect your diffusion locally if you're talking about a small molecule.
Thank you. I guess the other aspect is typically you're also looking for markers in the blood of some sort of measure of target engagement. Again, this is a bit more challenging with the inner ear. Have you got any comments on that?
I think there's plenty of people also here in the room that have looked at markers, everything from microRNA to protein markers, both locally and in circulation. And I think it's something that we still need to continue looking into. But yes, I think it's not necessarily just the isolation of the target from the circulation that's a problem here, but also the size of the target. A very minimal amount of tissue to release potential biomarkers into the blood. I would probably want to look more in the direction of functional markers of target engagement, similar to what you could do with EEG and or other imaging markers for CNS. What can you do with ABRs? What can you do with electrocochleography? What can you do with distortion products? What can you do with ASSR? Other ways of looking at how you're potentially dynamically interacting with the system might be a problem if you're looking predominantly at a autoprotective compound where the engagement only really is expressed longitudinally in the ability to avoid damage to the system, but other type of programs, if you have an acute modulation of a target that affects the output of outer hair cells or that affects synaptic transmission, you might be able to get to something there.
Thank you.
All right, thank you. Our next speaker is Dr. Gail Naert. She comes to us from Silcare, where she's the Chief Operations and Scientific Officer. The title of her talk, Partnering from Discovery to the Clinic, Silcare CBSET Integrated Auditory Services. Thank you, Gail.
Thank you. Just using a pointer. Okay. So thank you, Colin. I would like to thank the organizers of the PIHL Symposium session, and particularly you, Colin, you are the person who has made this happen. It's my great pleasure today to present you Silcare and CBSET Integrated Auditory Services. And partnering with us is the best way to support the development of your therapeutics from discovery to clinic. Silcare CBSET is a one-stop-shop partner for external innovation in hearing disorders, providing a full range of preclinical services from early screening to GLP autotox studies, and this covering all the preclinical package before submitting your IND or CTA. And in the last 10 years, we have the chance to work with various therapies, drugs, including small molecules or peptides, for example, but also gene and cell therapies, cochlear implant, and drug delivery device. CBSET is a US GLP-compliant preclinical research institute with more than 20 years of experience in regulatory translational research. And Silcare is a company dedicated to hearing disorders, founded in 2014 and in France. And in 2017, Silcare and CBSET partnered to enhance and develop their offering in auditory services. And this alliance puts together Silcare expertise with CBSET operational excellence. And Silcare CBSET has a huge lab capabilities with 2 different locations, one in France, in southern France in Montpellier, and the other one in the US close to Boston. I'm not going to detail all the equipment and the rooms that are listed here, but I would like to say that Silcare facilities in France are perfectly located to serve our European and Asian customers. On the other hand, our American customers can benefit from our facilities in Lexington, in the center of Boston Biosystem. In order to answer sponsor needs in translational preclinical research, we offer a full range of models. And as an in vitro models, we have the HEI OC1 cells, And on the other hand, we also offer cochlear explants from different species of rodents, such as mice and rats, and different type of protocol and models, as you will see later in this presentation. In addition, we work with the most used species in hearing research: the zebrafish, different strains of mouse, rats, guinea pigs, chinchilla, gerbil, but also with large species like the swine and the sheep. Over the past 10 years, we have developed several platforms with different techniques and models in hearing research. And I'm proud to work with an amazing team, highly passionate and competent, with strong background in neuroscience and otology. And today, in fact, we have several experts for each of these platforms that I will detail in the upcoming slides. Our first recommendation, as you mentioned, Jonas, you have to de-risk your drug development. And for this, we offer different screening models that can have, in fact, two objectives: the determination of potential of the ototoxicity of your treatment, and also the evaluation of otoprotective effect of your treatment. But whatever the objective, the use of these different screening models allow to de-risk your therapeutic development, and this in gaining time and money. We offer a screening model using this in vitro model, the HEI-OC1 cells, with a various different techniques. And this is suitable for when you have many, up to 100 conditions, or drugs or doses. And as a second screening test, we can do an in vivo screening with a zebrafish model. And for this, we have developed also different models for autoprotection with cisplatin and aminoglycosid, for example. And at the third model, which is more complicated, we have also an ex vivo model, so the cochlear explant. And this cochlear explant can be used for with different protocols, and enable you to have a more specific indication of ototoxicity, as we are able to see all the cochlear cells in this model. And also this model can help you to have more information on the mechanism of action of your drugs, and enable you to have the dose, the therapeutic concentration of your drugs. And when you have performed this screening test, you can do, for example, some tests in ototoxic models that we have developed using different ototoxic drugs. So, for example, the cisplatin, or the gentamicin, and the kanamycin. And as you can see here, these three drugs induce a well disorganization of the hair cells with a base to apex gradients. This is just an example. We have validated, in fact, different models and protocols, and here the focus is really to target the hair cells, but we are also able to evaluate fibers or AGN in this cochlear explant. And when you have achieved these screening models, the next step is to go in vivo and to test your drugs or your therapies in efficacy models. And for that, you need to be able to administer your drug and to reach the target organ, so the inner ear. And in fact, we are able at Silcare to perform all techniques of delivery, so of course the standard systemic delivery, but also all possible local delivery in the ear canal, in the middle ear canal, and also, of course, in the inner ear. Here, I would like to focus a little bit on the middle ear delivery. In fact, we are experts in transtympanic procedures in any species, and we have also developed other methods to deliver the therapy in the middle ear without affecting the eardrum. For example, we have developed a technique to perform the round window deposit, so with a retroauricular approach, which is a surgery. We are able to deliver a liquid solution or a specific formulation on the round window niche. But we have also developed two other methods, again with this surgery, to deliver as an acute or repeated administration using a transbular port, or with continuous infusion using a micropump, like an Alzate osmotic pump. And as you all know, if you would like to obtain a long-lasting release of the drug in the inner ear, after the delivery in the middle ear, it is preferable to deliver a specific formulation. And this is a type of services that we offer. So we have services for formulation design. So you can develop any type of formulation. It could be thermoreversible gel or microadhesive formulation, for instance. And the development of the new formulation adapted to your drug or your product is also highly relevant when you want to do auditory research. And in fact, up to now, we have developed more than 15 formulations which were successful in vivo. So I mean that, in fact, these formulations were tested for tolerance, and they did not induce any sign of ototoxicity. They were able to deliver the drug into the inner ear, and this at a therapeutic concentration. And for this, we have in fact performed different pharmacokinetic studies. This is really studies that we perform routinely at Silcare and CBSET. And we have huge expertise in inner ear pharmacokinetic studies, with most of the studies performed with transtympanic administration, as you can see. And we are able to sample in same animals different tissue, of course, all the tissue and fluids related to the inner ear, so the inner fluid, the cochlear tissue, and we can also sample the different other tissue, like the brain or the CSF. For the brain, as we have experts in neuroscience, we are able to sample any type of structures related to auditory pathway or to limbic system, for example, and we can do any sampling of peripheral organs and fluids. And when you have performed this first study, the next step is to do efficacy study, and for this you need to have a good evaluation of auditory function. And as any other labs in auditory field, we have all standard procedures, meaning that we have ABR measures, DPRE measures. We are also perfectly equipped with a different setup for noise exposure, which are well calibrated and validated. But however, what makes us unique is the fact that we have standardized this technique and provide high-throughput methods. And as an example, for the ABR, we are able to assess 8 animals at once. So meaning that we are able to assess on these 8 animals the left ear at the same time, and then we are doing the right ear. And this allows us to really reduce the duration of the study, and we are able to conduct and to assess many animals in a day. And in addition to that, we have also, for the analysis of the data, we have developed our own algorithm to determine ABR threshold and also ABR waves analysis. And this allows an accelerating evaluation of hearing in animals. And we are able to deliver, as I said, in a short time frame, a study, but also conduct several studies in parallel. Another main assessment in your efficacy studies is the histopathology analysis of the cochlea. And for that, we have all the technique in the field, so we do all the flat surface preparation of the cochlea, like cochleogram or ribbon synapses staining and counting. We also perform cross-section of the cochlea and H&E staining to assess if there is any sign of inflammation or any damage of the structure of the cochlea. We are also able to do nerve and SGN analysis. We are also equipped to do SEM or TEM. And in addition to these several analyses of the cochlea that I'm showing here, which are validated in many species, in rodent, but also in large species like in swine. We can develop any type of histological analysis on request of the sponsors. And regarding the translational model, we have validated in-house. We have a multitude of robust drug-induced hearing loss models. So we have cisplatin and aminoglycoside models. These models were developed as acute models, that meaning that we are administered the cisplatin or the aminoglycoside just once, which is a very good model to have a first proof of concept in this indication. But to have more translational models, we have also developed a cyclic model for the cisplatin to mimic what is performed in patients in clinic. And we have also performed chronic administration. So we administer the aminoglycoside daily during several consecutive days to mimic what is performed in the patient. And for the aminoglycoside, we can also combine the administration of aminoglycoside with an antidiuretic agent like the furosemide. And as an example, I would like to show you our acute model of cisplatin in rats. As you can see, an administration of cisplatin at 10 milligrams per kilo will induce an increase of ABR threshold and a decrease of DPOE level, meaning that we have a permanent hearing loss in this model, even if it's a very short model. And we have tested the only one drugs which have been validated for the moment, so the STS, and so the sodium thiosulfate, sorry. And we were able to totally reverse the cisplatin-induced hearing loss in this model. And regarding the analysis of the cochlea, as you can see, the cisplatin induced an increase, a hair cell loss in this model, and mainly at the highest frequency, so at the base of the cochlea. And this effect on the outer hair cells were also totally prevented by the sodium thiosulfate. By this model example, I would like to show you that we have developed robust models that can be reversed by drug administration, drugs that are known to be autoprotective. As other hearing loss model, we offer also a large range of noise-induced hearing loss model. So we have different type of noise exposure for this model, and with this noise exposure, we are able to induce PTS model or TTS model, so permanent hearing loss or transient hearing loss in different species. And this model were well characterized for the hearing level, but also for the signal transmission with ABR wave 1 amplitudes assessment, and also the counting of the functional synapse on the inner ear cells. And this was also completed with a full characterization of the tissue with a different technique we have on our histology platform to have a full characterization and a comprehensive study of this noise-induced hearing loss model. In addition to that, we have also models for tinnitus and hyperacusis, if you are interested. So, as you know, this model can be induced by the use of ototoxic drugs, but also by noise exposure. And I would like to emphasize here, as an example, the salicylate-induced tinnitus model, which is really a good model for having a first proof of concept of your therapy. This is a model which is very short in terms of duration. It's just a 1-week study. And we are able, using the gap detection, to show that after one administration or 4 days of administration of salicylate, we have the presence of tinnitus. And in the same model, we also assess, using the startle response amplitude, we assess the presence of hyperacusis, so a different startle sound intensity, and we can see that we have the presence of hyperacusis in this model. Then I would like to show you our platform for gene therapy. And if you want to work on gene therapy, you have to be able to deliver your drug in the cochlea. And for this, we are experts in the different intracochlear administrations. So we can do the post-semicircular canal injection. We can do also the round window injection with or without a PSCC vent. And we also do cochleostomy to perform a single injection as a bolus injection, but we can also do a continuous infusion by using a micropump like an Alzad pump to deliver at the base of the cochlea the formulation or the product. Here, as a first example, you can see images of a biodistribution study after a PSCC injection of 2 different AAV, which are commercial AAV. And you can see the typical result we get with these 2 AAV. In this picture, we show, in fact, a flat surface preparation of the cochlea, and you are able to see that we have a good distribution of this AAV, for example, in the inner ear cells, and we have an expression as a gradient from the base to the apex. As another example, we have also performed round window injection in guinea pig and in mice of 2 different AAV, again, which are commercial, and you can see the biodistribution of this study, of this AAV, sorry, which is also, here we have a base to apex gradient of the expression of this GFP AAV in the guinea pig, and in this one, in mice, we have an apex to base gradient of the expression of this AAV. And finally, for the gene therapy platform, I would like to show you one example of a study we performed in collaboration with Aposense, which is a company that develops specific siRNA. So in fact, this RNA have molecular nanomotors, which enable this siRNA to better enter into the tissue, or in our case, to better enter in the cells of the cochlea. We have injected this siRNA using a micropump, which were connected to the base of the cochlea, and we inject this siRNA. In fact, it was an infusion for 24 hours. And we make a comparative study of a commercial standard siRNA labeled with a size 3 fluorochrome compared to this construct with the M&M siRNA from Aposense, also with a size 3 fluorochrome. And you can see that the red signal is more pronounced when we are injecting the siRNA with the M&M, and mainly around the hair cells, and you can see that we have, again, a base to apex gradient of this expression. And Silkia CB-set also offer to conduct auditory studies in mini pigs, which is, in fact, a good alternative model to non-human primate model. On this swine, we have the capabilities to do all the different techniques and methods that I have just presented. So, for example, we are able to do ABR, as you can see here on this representative ABR thresholds. We can also do all the histological analysis, and here you have an images of the cytocochleogram we have done in a mini swine. And for the route of administration, we have also done transtympanic injection, but also intracochlear administration in the inner ear. And we have performed some study as well with the cochlear implant. And based on the expertise of our partner, CB-set, which work with swine for more than 20 years, we are also able to do other techniques and more related to cardiology. And the last but not the least services that we offer at Silkia CB-set is GLP ototoxicity evaluation, which, as was mentioned before, is required before submission of IND or CTA. And this GLP study is conducted following the guideline of the regulatory agency. And mainly, in fact, the ototoxicity will be assessed using ABR otoscopy by in-life assessment. And at the terminal assessment, we will do cochleogram and histopathology, so H&E staining on cross-section of the cochlea. And up to now, CB-set and Silkia has conducted many GLP studies using different routes of administration and including also, as you can see, local administration. And beyond our excellent technical capabilities that I just presented you in the previous slide, I would like to bring your attention to our outstanding skills in project management. So when you partner with us, you are sure to have, all along your study, a fluid communication, a regular study update, and respect of the deadlines. And our track record achievement with more than 300 programs delivered for almost 100 sponsors and our involvement in more than 10 grants, our achievement confirms this commitment to excellence and our commitment to the company and the people we are working with. And to finish, I would like to show you some of the papers and books we have published these last years. And now we are looking to publish much more, building on current and future collaboration. So I hope I have convinced all of you that Silcare CB-SET is a perfect partner to fast-track your therapeutic development for hearing indication. So thank you for your attention, and see you at booth 1.
And we have time for a question or two. I actually have a question for you. You talked about all of the different species that you're able to work with and all of the different models that you're able to use. Do you have any guidance for the audience on how you would go about selecting the best species or the most appropriate species for their studies? Does it depend on indication, drug? What are the factors that go into selecting those animals and models?
So thank you, Colin, for the question. To have guidelines to select the best species, in fact, we need to know which is the indication, as you said, but we need also to take to consider the type of mechanism of action you have and the type of therapies you are doing. For example, if we want to do gene therapies, it's not possible to do, for example, PSCC injection in guinea pigs or rats due to anatomical differences. So we will select for sure mice, and also we will select the mice because we need to have transgenic mice to assess the efficacy of the gene therapy. So it depends of the product we want to test and the indication. For example, if we want to do cochlear synaptopathy, it's preferable to use the mice and the rat, and we have developed this model in both species. And in fact, we will propose to do two types of study, one in mice and one in rats, to have robust results of your product that is targeting cochlear synaptopathy.
All right, let me invite our next speaker, Dr. Rolf Holm, to join us. Dr. Holm comes from RNID, the Royal National Institute for the Deaf, where he is the Director of Research. He's going to shift gears and talk about towards a core outcome set for hearing therapeutics. Please help me welcome Dr. Holm.
Okay, thank you, and it's great to be here. So what I'm going to do is give a bit of an update on some work that we've been doing that I hope will lead to a core set of outcome measures that can be used in clinical trials of hearing therapeutics. But first, I'll just say a quick thing about RNID. So RNID is the UK's national charity, supporting more than 18 million people in the UK who are deaf, have hearing loss or tinnitus. We have a big focus on accelerating the development of treatments, and we do that by funding research and through our hearing therapeutics initiative, really helping to create partnerships and bring the research community together around key challenges. So one of these challenges that we've been focusing on is outcome measures. So currently, there's a real lack of consistency in the use of outcome measures in clinical trials of hearing therapeutics. And there's little consensus around what outcome domains should be measured, how these domains should be measured, or what represents a meaningful change within these domains. There has been core outcome research in other areas, but it's tended to focus on devices, and there haven't been any studies focused on sort of drug, gene, or cell-based therapeutics. And previous outcome research tends not to have involved the healthcare regulators as a key stakeholder. So this can create uncertainty amongst regulators, but also for developers and investors in new therapeutics. And obviously, it makes it really hard to combine and compare data across clinical studies. So we think there's a real need to develop a core outcome set for clinical trials of hearing therapeutics. This outcome set would include domains. So what are the kind of the key aspects of hearing and quality of life that are important and meaningful to measure when we're evaluating new therapeutics? It would contain recommendations as to what instruments we should be using in order to measure change within these outcomes, and guidance around what is a meaningful change within these domains. This would represent a minimum core outcome set recommended for all clinical trials, but clearly different domains would be added on for different types of hearing loss or different patient groups. I think it'd be really important to develop this core outcome set as an international consensus involving people with hearing loss, with regulators and payers, with clinicians and those delivering clinical trials, and companies and academics developing therapeutics. And that's important because we need to get buy-in from everybody who's going to be using this core outcome set in the future and making sure that it's meaningful. So there are standard and well-used methods for developing core outcome sets. So OMRAC is an initiative that's really pioneered the development of outcome sets in the field of rheumatology. And COMET is an initiative that's really championing sort of best practice research in core outcome sets. And has been used widely in other fields. So these approaches tend to follow sort of two phases. The first phase is about understanding what needs to be measured, what's important and what's meaningful, and developing consensus around that. It tends to start by defining the scope of the core outcome set. So what's the condition, what's the population, what's the intervention, identifying the key stakeholder groups, carrying out a systematic literature review to identify existing outcomes and gaps, qualitative interviews to identify what the gaps might be in the outcome domains, and then consensus methods with the stakeholder groups, such as Delphi surveys, in order to agree what's the most important outcomes to include. The second stage is focused on agreeing what are the most important measurements to use. And again, this follows the same kind of process, identifying what instruments are currently being used, assessing their quality and feasibility, and a consensus exercise to understand what's the most important. So our progress to date has been focused on pulling together a group of companies with a shared interest in establishing consensus around outcome measures, and who would be willing to share the cost of this project. The initial project is focused on the first stage of the process, so defining what are the core outcome domains that are important. The plan is to have a broad scope, so it would focus on all types of hearing loss, it would focus on adults, and importantly, it would focus on drug, gene, cell-based therapeutics. We're preparing to run an open call for proposals from groups interested in delivering this project. proposals we'd expect them to involve international groups of stakeholders for the consensus process. We'd want the project to deliver core outcome domain sets that are relevant to UK and European and US regulators. We'd want the project to align with existing outcome measures studies. And importantly, we'd want it to be delivered by an experienced team in developing core outcome sets, experience of auditory outcome measures, and of course access to those international networks in order to deliver the project. We've started talking to the regulators. They're very interested in this project, very supportive, and they're keen to engage with the group that will deliver this project. So if you'd like to get involved, please do get in touch. We're currently seeking additional industry partners who might be interested in joining and supporting this project, and we're keen to identify core outcome set developers who might be interested in delivering this project. And obviously, when the project gets going, there'll be opportunities for everybody to get involved in the consensus process as a stakeholder. So I just want to finish off by reminding everybody about RNID's Hearing Therapeutics initiative. So through this initiative, we can help connect you to the experts and the knowledge that you need to develop treatments. We've got over 250 members, so please do sign up if you want to join our network. And just to give a heads up, this November, on the 13th of November, we'll be running an in-person conference in London to bring everybody together again to explore the opportunities around hearing therapeutics. So please kind of save the 13th of November. If you scan that QR code, you can sign up to our network and we'll let you know when the registration process opens. Okay, thank you.
All right, we have 20 minutes for a discussion prior to coffee break. I know I have lots of questions, but I want to give the audience an opportunity to ask questions. Well, I'm going to jump in and invite people to come up to the microphones with their questions. Ralph, one of the things that strikes me about putting together an international effort is the language issues. We talked, you heard Jonas talk about speech and noise being a key issue, but thinking about speech and noise tests, language could be a barrier. And if you're looking for international consensus, how do you think the group might approach language challenges within an international consensus framework?
So that's a good question, and I'm not sure if I'm the expert to answer that. So if welcome for any other people who might have a better idea.
Yeah, thank you.
Yeah, maybe just a comment on your question. Of course, this is something that has been a problem in many international studies so far. But actually, there is, at least for the sentence comprehension, there is the international matrix test, which is available in almost all languages, including most European languages. For English, it's even British and American, and this is well calibrated towards these languages and really makes this test comparable within international trial efforts. I mean, it's not, monosyllables and quiet. For that problem, we don't have a test yet, but I think for a sentence test in quiet, for the SRT and speech and noise, the international matrix test is applicable.
Thank you. The other test that is emerging, and we have not used it, I don't know that much about it, there hasn't been a lot published with it yet, but I know there were several presentations last year about the ACT test which has become available and looks at some of the temporal changes in auditory signals without using speech signals in order to try and come up with the language agnostic test. And so I think it'll be very interesting as data emerge for that particular test as well.
To ask two panelists a question. One, Gail, on your example of cisplatin ototoxicity, was the STS delivered like it is clinically?
No, we were not able to have it, so we use a commercial one.
Thank you. And then my next question is for Ralph. Is the idea that any of the QOL instrument would be clinically delivered, meaning a clinician would do the Q&A with the patient, or would the patient do a patient-reported outcome measure?
I think what's sort of the sort of driving thing behind what we're thinking is having, as you say, that more patient-reported sort of outcome measure. So you know, we need to talk to patients and talk to clinicians and understand like what is the most important impact that hearing loss has that our treatments in the future need to address.
Yeah, because just experience, when we were doing our Meniere's trials and patients report tinnitus, and we used the TFI, and we focused on question 2, the visual analog scale, we decided not to ask the patient to lateralize which ear was the most affected, because in a lot of our patient discussions, patients would say, well, my left ear is most affected. Well, then a month later, I hear it in my head. And then a month later, it's in the right ear. So I think one of the difficulties with hearing loss, especially acute and chronic disease, asymmetry is going to have a huge impact. You know, when we think about Meniere's sudden sensory, obviously, and the episodic nature of that, but even some people with early acute noise-induced hearing loss, the asymmetry, acute ototoxicity, the asymmetry, it's something that we'll have to at least think about.
Good. Ralph, I think this is a really great idea. I wondered if you had considered expanding the scope to also think about how to best stratify people for clinical trials, because I think it's really going to be relevant, the outcome measures that you're talking about are going to be relevant to who you pick to put into a particular clinical trial, depending on the target and the particular diagnostic tools that we've got available. So I just wondered if you'd considered expanding the scope to stratification for clinical trials as well.
Yeah, so when we were thinking about the scope, initially we were thinking, you know, do you need to do these kind of exercises for every single type of hearing loss? But it would then become, you know, quite challenging, and where do you start? And so the sort of advice we got from experts was it's best to take it quite broadly to start with, and then there'll be sort of subdomains that might sort of pop out. I mean, I think what you're describing is like really important, and we definitely need measures that sort of predict what pathology is sort of causing the hearing loss. I'm not 100% sure whether this work will be that precise, whether it's going to be more that kind of patient, sort of what's important to the patient rather than the kind of the research kind of question. But yeah, maybe we can talk about a bit more.
Hi, this is at risk of maybe repeating that question, but slightly different slant. So we've got two companies who have taken forward assets into the clinic, and one of the challenges early on is to identify what your position in the market is going to be, and therefore align your biology to a patient group specifically in order to maximise your chance of seeing a signal in your early studies. So I wonder if you, Jonas and Jonathan, you can maybe comment on some of the challenges you face there.
I think I'll defer to Tim on that question because I think it's coming up in our presentation.
Okay.
So I don't want to steal the thunder of the next talk from our side. Probably Jonathan too.
Yeah, for our most advanced indication, Meniere's disease, there are academy criteria there. I'll present on that later in this session and some of the improvements that we've seen in pure tone audiometry, speech discrimination, tinnitus using the tinnitus functional index. We've pulled in 3 other PROs, the Vertigo Symptom Scale short form, the Dizziness Handicap Inventory, and a visual analog scale of oral fullness. So we tried to pick the measures that are most clinically validated and correlate to the disease. Obviously, are they sensitive to improvements in treatment, since no one's really been successful other than Fennec Pharmaceuticals using STS in a pediatric population. That was obviously achieved using ASHA criteria for ototoxicity because some of the patients were so small and ABR was used. There were no speech discrimination tests or data available from those studies.
Jonas, I have a question that maybe you can talk a little bit about. You shared a lot about the regulatory process with us. I guess if you could talk about animal data, you talked about how the FDA no longer requires use of animals for safety tests, but you need to justify whether you use them or not. Could you talk a little bit about how much efficacy data is necessary from animals and whether that's a major part of the packet or if that's a lesser concern within the regulatory packets to help people think about how much is really needed before they move ahead to human testing?
Well, I think legally speaking, it's not required, but everything comes down to risk-benefit assessment for all of this. So the more risk you have, the more likelihood of benefit you have to show. It really comes down to, I think, as so many other things, an individual evaluation on the particular program. But to a large extent, I think the benefit assessment is more of an internal confidence from the person developing or the entity developing the compound, and to a large extent also your financial stakeholders. Is anybody likely to put in the money to carry out the risk assessments? Is anybody interested in putting in the money to do all the manufacturing, all the other exercises that you need to do to get into clinic? So if you are confident from a relatively small data set that this has a likelihood of working, you're still going to have to generate all the risk data set to underpin the fact that it's not going to be harmful. And in the end, I think the regulatory authorities are less concerned about whether you're wasting your money on this, as long as you're not putting anybody at risk doing so.
I think the first part of your answer was that what often surprises people, that it's not necessary. Jonathan, do you want to say anything about how you've thought about how much animal data is necessary before you move ahead with a clinical indication?
Yeah, just reiterate what Jonas said. There's no requirement to show proof of concept in animals. They will ask about the rationale of why should we expose a patient to your drug. And it's nice to tell a story, but the rest of it is pretty rudimentary. CMC, farm talks, first-in-man studies. It's not sexy science. A lot of it isn't really scientific. In terms of whether animals will no longer be required, yeah, I think that's really a fantasy. I can't imagine a regulator being even open to the idea that you haven't tested this in animals, especially if they look at the pharmacologic class and if they see something else in development. Like, you know, we know of some of the drugs now that are used as GLP-1 analogs. There was a little hold on the field a long time ago when some of these tumors came up in some of these mouse studies. They paused everybody to address that, and that's why there are warning labels for MEN syndrome and other related neuroectodermal tumors. So I think if you have a clean drug, it should be okay, but time will tell when we don't have to provide GLP tox studies.
I think it's also to what you say, Jonathan, a bit of a self-limiting exercise, right? In order to convince the regulators that you don't need to carry out the classical studies, you first have to invest a lot of effort, and not just for your own candidate, but a number of other candidates and reference compounds to demonstrate that the other assay is predictive and will eventually generate the same kind of data that they're comfortable with from the animal models. So if you don't have some degree of guarantee or some degree of agreement that this will lead you to not have to do it without doing it anyway, who's going to spend the time and the effort to generate this on a large scale? That's probably the big question right now. So I don't see it happening in the near future either. It may be something that accumulates over time, maybe for very particular populations where there's a high need and a limited risk, particular candidates, you start building this, but you're up against decades of historical data that validate which models are currently predictive and not predictive that you kind of have to replace. As Donald says, that the mandate is technically dropped from a legal perspective, but the regulatory guidances remain the same. And I say, anything is up for discussion if you believe that you have a case for exemption, right? But that's on a case-to-case basis.
Hi, I'm David Rosenberg from 8th Nerve Consulting. I have a general question for the entire panel. I was wondering, what are the biggest missteps or mistakes that you've seen companies or groups make in the process of drug development? Dr. Deerfield Johnson, I know you mentioned having multiple leads as being an especially important point. I was wondering if there's any other pearls that you could share with us. Thank you.
I mean, I don't know if there's any biggest mistake, but I mean, anything that's uncertain puts your development program at risk, right? Whether it's an uncertain dose prediction or, I mean, I think the things that have been identified as really key, and that's not by me, but by the Pfizer's Pillars of Survival reviews and all these kind of things, is you need to be able to measure meaningful change. You need to have a population that is adapted to what you're trying to do, and you need to ensure coverage of the target for a sufficient period of time. So if you feel confident in all of these things going into your clinical studies, then you're probably in a pretty good place. But if you're uncertain about any of them, then you're taking risks. I mean, they also clearly identified these factors that play into really the big open question is, can you say with a degree of certainty after running patient studies that you have robustly answered the question about whether something works? And I think they identified that a good chunk of studies, if not more than 50%, could actually not confidently say, yes, it works, or no, it doesn't work, but somewhere in the middle, which is not something that's very confidence building for a phase 3 study eventually, right? What are you confirming if you're not sure if it worked in phase 2 already?
I think I've seen over the many years that I've been coming to this meeting, two significant mistakes. And one would probably be in neurodevelopmental signal transduction. It's a nice idea to try and recapitulate development in an adult system to regenerate it. But I think many of us who study that know that in adult animals, well after a lesion, that those signals are not robust. So advancing those into clinic, well, they failed. The other is more clinically related. I think injecting across the eardrum of a young adult, an adult repeatedly, even once a week, times 3 to 4 weeks, is well tolerated. I think trying to do that in an aged person who's 75 years of age, that last injection may not heal so well. Oftentimes what we hear in some older patients is that when they come back for their third or fourth series of dexamethasone injections, especially if the patient's older, that hole hasn't healed, and it can be a nidus for infection. So, yeah.
Go ahead.
Since you're not talking about the regeneration, Jonathan, I'll add one thing that maybe can be illustrated in that direction, but actually applies to everybody, is not subjecting your drug candidate to a tough enough examination preclinically. You can put yourself in sort of positive conditions to see that something works, and I think that's what you're alluding to, Jonathan, with the regeneration. If you're testing it in 2-week-old mice, there's a lot of things that can work that won't work in an adult animal, even not even a human. But there are other things that maybe could be taken into account there, like how long are you waiting from deafening to regeneration? What kind of age of animals are you using in the first place? How long do you wait for your readouts? All of these kind of things. This all complicates your models and your studies a lot. You might need to wait for an animal to grow to an older age, then wait a couple of months even more, and then eventually try to treat something. This is a gamble, of course, because no certainty that it's going to give you an interesting and positive result. It's going to take time, it's going to take effort, but in the end, if it has to have a meaningful chance of working clinically, it's probably still less money, less time, and less effort than running a futile clinical trial. So critical experiments as opposed to confirmatory experiments.
I have one more question before we have our coffee break, and I see coffee back there, so I imagine people will keep their answers a little bit short. But this idea of risk tolerance has come up several times, and I wonder if members of our panel could comment on how risk tolerance might change over time as you think about academic labs and startup companies and small but established pharmaceutical companies, and then moving towards larger pharmaceutical industry. How does the tolerance for risk potentially change across that spectrum?
I don't know if it's linear like that. I'd rather say it depends. I mean, you have certain luxuries if you have multiple programs or multiple candidates or already have revenue. All of these kind of things can play an influence. On the other hand, many of the larger organizations are change averse. So taking a risk on a new field altogether might not be in their DNA.
All right, we'll take a break here. We have 20 minutes set aside for coffee and networking. So we will start again at 3:15 with 3 additional speakers. And then we'll have additional discussion opportunities and move into our social hour. So please enjoy some warm coffee. It's a little chilly in here. Hopefully it warms you up. HCE programs.
For sure.
They were receptive to that.
And at some point, I'm sure some people, things will change. They have changed so many times. I mean, just yes, I am. Yes.
You know, we're planning to just shut it down. They really felt it was done and wrapped up and that they weren't the right group to rejuvenate it because it had been so much baby and we're gone. Nobody ever was excited. So, yes, if there's anybody in this room who is a young person who wants to move on, you need to contact me. I don't have to do it. When
Courtney Hecht is here. Have you
yeah,
talked to her?
yeah.
I don't know if it's the right time, but he's
No,
energetic.
it's great because they originally was going to send anyone to represent the
And he's the only one?
Well, then once people who signed up, we don't have 200, it was a huge registration.
No, that's good.
So they decided they really should send.
Okay, good.
So that's how we got it.
In my lab.
So you're postdoc? I was going to ask. And what's your topic?
You're still looking for one?
Found everything. The other problem.
Oh, really? Yes. Okay. Awesome experience. I think Laurel's interested in anything like this.
Ability All right, if I can get everybody to start moving back towards your seats and get my panel to make their way back up front. While you're making your way to your seats, I want to thank our sponsors again. Without their support, we wouldn't have this event, so please make sure that you thank them as you have the opportunity to network with people over breaks. Our next speaker is Dr. Amanda Henton, who comes to us from Turner Scientific, where she's the Chief Scientific Officer. She's going to talk with us today about preclinical contract research services, advancing hearing loss therapeutic development through translational animal models. Please help me welcome Amanda.
Okay, so thank you, Colleen, for the kind invitation to speak today and for organizing this symposium. So I'm happy to talk to you all today a little bit about Turner Scientific and our contract research services, and specifically how we can help you advance your preclinical pipeline. So in recent years, there's been a lot of new excitement and energy in the inner ear disorders space with some recent clinical trial successes. We heard last year about the otoferlin trials in humans, and also about Fennec Pharmaceuticals' FDA-approved PEDMARK for cisplatin-induced hearing loss. However, while we have these two new successes in the field, we still have relatively few therapeutic options for patients, especially when compared to other sensory systems, such as the visual system. And I think that there are a few key reasons why, and a few challenges that still remain in the field. One of the biggest challenges, I think, is really an incomplete understanding of cochlear pharmacokinetics and drug distribution, especially in the human cochlea. There's also the technical difficulty of local drug delivery. It's often, as a few speakers have mentioned so far, really difficult to reach the target organ of the cochlea. And further, I think the field has really been hampered by poor translation from rodent models to humans. And this, I think, really the problem is underlied by a lack of validated and accessible large animal models. However, at Turner Scientific, we really aim to be your outsourced R&D partner and aim to circumvent some of these issues. And how we do this is really in our expertise in inner ear drug delivery, as well as cochlear pharmacokinetics and computational modeling that I'll chat about shortly. And as a CRO, we have much higher throughput capabilities than contracting with university labs or other things. And we also have no claims of intellectual property or publication rights. So we're really invested in your success in developing your pipeline. Further, one important feature of translating studies from the basic lab into the clinic is independent validation. This is often very important for investors to see that results obtained in academic labs can be replicated by an independent third party. And at Turner, while I think we have plenty of really excellent ideas for new inner ear disorder therapeutics, we're not developing our own clinical pipeline. So our interests are really in your success and your pipeline. And further, we have a scientific team with decades of experience in auditory research coming from academia and industry, with expertise in drug delivery and disorders such as tinnitus and hyperacusis that can really partner with you from early discovery work all the way to pre-IND filings. And so for the remainder of this talk, Jonas really laid out a really great framework for preclinical and clinical trial success. And I'd like to discuss more what we think are really crucial steps for a successful preclinical pipeline. And these are not an exhaustive list of our services, but to highlight a few key stops on this roadmap for the preclinical pipeline. We think a really important starting point is really understanding the pharmacokinetics and distribution in the cochlea of your therapeutic. And we can achieve this through precise PK studies and sampling and computational modeling. Additionally, we have a full sort of range of in vivo safety and efficacy testing with validated disease models such as noise-induced hearing loss, cisplatin aminoglycoside toxicity, and others. And we also have a full range of key safety and efficacy readouts that are often required by regulatory bodies, such as audiometric and behavioral testing in vivo, and as well as ex vivo cochlear histology and imaging. So one thing that I think really makes Turner Scientific a great partner, especially in early discovery and PK work, is the fluid sim model. So this was developed by our director of cochlear pharmacokinetics, Dr. Alex Salt. And what this model really aims to do is to understand the distribution of compounds throughout the cochlea. In this panel here, you can see just an example. So you can enter in key properties of your therapeutic, such as molecule size, polarity, things like that, and also the host species and the method of delivery. And you can actually model in real time what fluid compartments and over what time course that drug is expected to distribute. And Fluid Sim is available in 7 animal species and in humans. And we think that this has several key advantages in preclinical research. Specifically, Fluid Sim can help you design better experiments in that before we even get to the in vivo stage, can really help aid in choosing the best dose levels, volumes, and time points for PK studies based on the predicted computational modeling. And furthermore, a better understanding of the molecule and the cochlear physiology can help you understand which compounds may be better suited for systemic or local delivery based on their residency times. But I think a really key feature of FluidSim is that it's a helpful resource in translational research. In the field in recent years, we've seen several drugs that show really remarkable efficacy in rodents, but when translated to the clinic, really fail to meet any clinical endpoints. And I think an important reason for this is that they're very different sizes of the cochlea. When drugs may be locally applied to a smaller rodent cochlea, it may distribute very well and show great effectiveness. However, when a small molecule may be delivered to the basal turn of a human cochlea, it may be rapidly eliminated to vasculature, and due to the much larger size of the human cochlea, that drug may never distribute to the apical regions of the cochlea responsible for low-frequency hearing. That's especially important for speech intelligibility and really important clinical outcome for patients. And all of this together to say that Fluid Sim can really lead to significant cost savings and helping you design higher quality studies earlier in the process. But not only is it useful early and in the design phase of the study, it's also really useful in interpreting results from PK studies and deciding next steps and strategies when translation to humans. So in addition to modeling, at Turner Scientific, we have great expertise in cochlear PK sampling. As some of you may know, sampling of the inner ear fluids is a highly specialized technique, whereas the parallel volumes of rodents can be quite small, so sample collection and very accurate and precise collection is necessary. Otherwise, there can be really distorted or uninterpretable data. We can also have more specific sampling techniques. This was also pioneered by Alex Salt and Jared Hartsock, both on staff at Turner, where we can do sequential sampling. And this allows for the quantification of drug gradients along the cochlea, so you can really understand whether your therapeutic is actually reaching the target. And that's really one of the core principles of pharmacology is that you're getting a therapeutic concentration at the target organ. Additionally, we also offer cochlear PK consulting services to help interpret fluid sim data or results from PK studies. And I should say we can do cochlear PK sampling in a wide variety of species, from small rodents, and we're also developing these methods in larger animals. So once you've established that you have target engagement in the cochlea, it's really important to have standardized safety and efficacy measures. And so some key outcome measures that are really specialties of ours at Turner are the more traditional cytocochleogram. This is including hair cell and synapse quantification that you can see an example of in the left two panels. As well as paraffin-embedded H&E sections that are really important when delivering drugs to the cochlea to demonstrate safety and efficacy. And we also partner with certified histopathologists to do these reads. We can also do quantitative assessment of gene transfections in different cells of interest, as well as cryosections, other frozen sections. And I'll speak a little bit later, we've also developed really cutting-edge 3D light sheet microscopy that's really useful in imaging the entire intact cochlea. In addition to our ex vivo histology methods, there are a few safety and efficacy tests that are important in vivo, such as the auditory brainstem response and the distortion product otoacoustic emission. We also have a full range of behavioral testing, from tinnitus and behavioral audiometry to cognitive assessments, as well as vestibular and motor coordination tests. And again, we have a variety of normative data sets for our noise-induced hearing loss models, as well as chemotherapeutic or aminoglycoside models. And to speak more about some of the translational issues in the field, really it goes back to having validated animal models for inner ear research. We have several models that can be suited for different therapeutic indications and different stages of development. While we think that computational modeling is an excellent first step, we also have cochlear explants from mice and rats with spiral ganglion neural cultures. And this is a really good model that allows for low cost and high throughput drug screening. We also have in-house breeding of mice available of wild type and also mutant strains in rats. We also have an active guinea pig breeding colony, which we use extensively for PK studies because they're a really great rodent model because they have a relatively large cochlear volume to body size. So it allows for these sequential sampling techniques and understanding in a small animal of the pharmacokinetics in the cochlea. And our large animal model that we've been developing for more than 2 years now is the miniature swine model. We use Sinclair nanopigs for device gene therapy, as well as toxicity, safety, and behavior studies. And I'll chat a little bit more about that later. But the other half of our business is really interested in the sensory experience of the animal. And so we make and sell the sensory sentinel systems. And these are systems that can monitor 24 hours a day in animal housing and laboratory spaces for noise, vibration, temperature, light, humidity, and staff activity. One of the biggest issues facing biomedical research is the reproducibility crisis. And we truly think that one of the biggest causes for this are these sort of unseen variables, such as ultrasonic noise or vibration, that can cause stressors or clear confounds in auditory research studies. So beyond creating more controlled environments for our studies, we also think that excellent science really begins with top-notch animal welfare. And all of our facilities are monitored with the sensory sentinel systems. Additionally, we've received ALAC accreditation at our main site. So, one of the issues traditionally facing auditory research is bridging this gap between rodents to humans. And traditionally, the large animal used in auditory studies has been non-human primates. But there are several challenges to using non-human primates for inner ear studies. And these being, they're incredibly cost prohibitive. In recent years, there's been even larger and larger availability issues and lead times. And because of these first 2 points, this often leads researchers to make tough decisions and often conduct underpowered studies where we may be missing really important results. Additionally, in recent years, there's increased cultural sensitivity to their use in biomedical research. And even regulatory bodies are encouraging companies to use alternate models and to only use non-human primates if absolutely necessary. But besides all of these points, we think that perhaps the non-human primate might not be the best model for some inner ear studies because their cochlear size and volume is not well matched to the human cochlea. So instead, we think that the Sinclair mini pig can be a much better alternative to non-human primates in hearing research. And there are several advantages specifically of the mini pig in that they are readily available and ethically sourced. Sinclair Bioresources is just about an hour and a half away from our Jacksonville site, and they're only a fraction of the cost of one non-human primate. So this really enables studies to be more appropriately powered and also to be able to do more studies. And they also, since they're used in agriculture, overcomes many of the cultural and ethical concerns of their use. Additionally, they're very docile animals and easy for behavioral training because we think that really the next stage of hearing therapeutics research, once we address some of the issues in the cochlea, is understanding the central effects of these drugs. And so we're actively developing several operant behavioral models for sound detection and for pitch discrimination and other things like that. Furthermore, their hearing range and cochlea size is much more similar to humans than non-human primates, cats, or guinea pigs that are also commonly used. And while their use in auditory research is relatively new, their use in regulatory experiments or experiments for use in regulatory submissions is not new at all. For the past 20 years, swine have been used in cardiovascular research and dermal research and are well accepted by many regulatory bodies. So one question you might be asking is, how many is a mini pig? Well, the Sinclair mini pig is generally about 20 kilograms at full grown. And while this may not quite seem as many, it's certainly larger than many non-human primate species used, but not quite as large as a domestic pig. And we think that this is really ideal for safety and toxicity tests because they have a blood volume that's much more relevant to human work, but not a body size that's so large that requires so much drug or test article. And the Sinclair mini pig has been progressively downsized through selective breeding over several generations, and we think are the ideal size for safety and toxicity testing. But back to the mini pig cochlea, some really elegant work by Adel Moadi and Turner's Alex Salt looked at the cochlear dimensions of the domestic pig. And as you can see in panel B, the domestic pig's scaled tympani dimensions are much more similar to human than the cat or even the cynomolgus monkey. And this is important, not only for the reasons that we've talked about in PK studies, but also their cochlea is more human-sized, so it enables studies using devices, cochlear implants or others, that are sized appropriately for the human. Whereas doing these experiments in rats or guinea pigs or cats or sheep often involves having devices that are scaled down for the human size. And regulatory bodies really like to see animal testing with the actual device intended to be used in humans. And the domestic pig is now integrated into the FluidSim model, and we're currently working to refine the segmentation and the dimensions of the cochlear compartments for the mini pig, and hope to have that integrated soon. And I should also say that the cochlear volumes being much more similar in pigs allows for more accurate PK studies. We can do round window injections, and we're also working on other sampling techniques in the mini pig that we think could really help bridge that translational gap between humans and understanding how much of your therapeutic could be reaching the target tissues. In addition, we have done a lot of work over the last 2 years to develop some really advanced imaging techniques for the miniature pig cochlea. And the left is just a quick video of a miniature pig cochlea. In green, we have hair cells labeled with myosin 7A. In red, we have spiral ganglion neurons labeled with neurofilament. And we think that light sheet imaging offers several advantages over more traditional cochlear histology methods. With light sheet imaging, we are able to keep the entire cochlea intact and use clearing methods to image the intact cochlea. And this can be really important for understanding if you have a gene therapy, what cells and structures you are getting expression in, especially if they are structures that are not in a traditional whole mount preparation. And also very important for device studies of you can image leaving a device intact and really understand the biological consequences of that. And on the right, you can see we've done some work to segment the mini pig cochlea, and we're working to get accurate dimensions to be able to be included in FluidSim. But I should also mention that the very same markers and antibodies that we use for rodent studies in mice and rats actually work very well in miniature pigs. So it's really a good thing for continuity of studies that you're able to use many of the same markers. These are just a few examples, but we've also looked at support cells, SOX2 labeling, which also works really well in the mini pig. And here's just an example of a cross-section to give you an idea of the level of resolution we can have with light sheet imaging to be able to resolve individual hair cells as well as auditory nerve fibers. And while we think light sheet imaging is especially advantageous for the larger size of the mini pig cochlea, we're also able to do more traditional cytocochleogram analysis and count both hair cells and synapses with a whole mount dissection technique, as well as cryosections and other methods. And in the miniature pig, we have developed our ABR techniques. These are just a few quick examples of an air conduction and bone conduction ABR. Bone conduction, we think, can be especially useful for studying disorders of the middle ear or where there's ossicular chain disruptions, as well as traditional air conduction ABR. Also, we're developing a noise-induced hearing loss model in the miniature pigs, as well as a blast-induced hearing loss model. Miniature pigs, because of their skull size that's similar to humans, are often used in TBI research with cognitive assessments, and we think that there's a great potential to use the miniature pig model in blast-induced and noise-induced hearing loss. And to finish up, I'd love to talk about some of the really exciting future directions for both Turner Scientific and the miniature pig model. In August of last year, we were awarded an SBIR grant from NIDCD to specifically help validate the mini-pig model for hearing research. So in this grant, we're not developing a therapeutic, but instead refining techniques that will allow us to generate normative data sets and accelerate work that sponsors would do with us. A few of these key aims of this NIH grant are to do some cochlear paralymph sampling experiments with apical, round window, and canal sampling so that we can really better understand the PK properties. We also aim to optimize ABR and DPOE measurements. And importantly, we are developing an operant sound detection task. As there's been developments in understanding cochlear pathophysiology and therapeutics addressing that, I think really the next important questions are understanding what are the central consequences of this. Sure, with therapeutics, we may see improvements in ABRs and DPOE, but how does that translate to speech perception and pitch discrimination, things that are all really important for patient outcomes? So we are developing a touchscreen task where mini pigs will actually swipe their nose on a touchscreen to indicate that they hear a sound. And beyond being very cute, we think that this can be further developed to do understanding tone and noise tasks, or perhaps even tinnitus and hyperacusis testing. So we're really excited about the future of the model and have some exciting news about Turner Scientific as a whole. We've recently expanded into a 60,000 square foot research building on a 9-acre campus. So this really enables us to increase our capacity for both large animal and small animal work. So I also want to thank the scientific team at Turner Scientific. Many of us are here. We'll have a booth outside of the poster hall tomorrow, and many of our consultants and on our team are here. So if you have further questions, please feel free to stop by and talk to us, and we'd love to talk about how we can help advance your research. Thank you.
We have time for a question or two. The question was, how long does it take to train a pig?
That is a good question. We're kind of in the very early stages. But as far as behavioral acquisition, a couple of days and you can get the animal acclimated to touching the screen. So we're not quite sure how that timeline will map on to more complex tasks, but they're remarkably intelligent. They can learn their names and kind of basic commands. So really excited about the future of behavioral training.
Just a quick question. What's the lifespan of the pigs?
The lifespan is several years, I believe. For most of our studies, either AAV or device studies, we begin with juvenile pigs, somewhere between 4 to 8 months old. However, as far as longitudinal studies go, we've conducted studies anywhere from 2 months in duration up to 6 months post-surgery.
I have one more question. You mentioned developing noise models. Do we know anything about the relative vulnerability of the mini pig compared to rodents and macaque monkeys and humans, where we see a huge difference in vulnerability?
Yeah, so that work is still really in the early stages. There was some work presented at last ARO with blast exposure and some noise-induced models. And I think the data suggests they may be more similar to primates, to where more intense exposures may be needed to induce hearing loss. But in our grant, we plan to test several different noise intensities and duration and the effect on ABRs.
Thank you. All right, our next speaker is Dr. Tim Balke from Acusia Therapeutics, where he is the Chief Executive Officer, Chief Medical Officer, and a Managing Director. He's going to be talking to us about ACOU 085, a locally administered KV 7.4 enhancer for the prevention and treatment of acute and subacute forms of sensory neural hearing loss. Please help me welcome Tim. Thank you.
Good afternoon, dear colleagues. First and foremost, I would like to thank Colleen for the organization, but also for the invitation, and in order to have the opportunity to actively participate. Now, when I looked at the program earlier this morning, I realized that probably today, and you've heard that from Colleen, I will not get the gold medal for the most catchy title of the presentation. It's pretty clumsy, but in essence, what I'll try to do over the next 25 minutes or so, give you the background, but also some of the details of the clinical phase 2 hearing loss program with Bimokerna, which was until very recently only known as ACO 85. Now, many of you, if not most of you, probably don't know Acusia Therapeutics. So who are we? Actually, we are a spin-off company. We're based in Germany, in Tübingen, in the southern parts of Germany, spin-off of the ENT clinic of Tübingen University. One of the 3 founders of the company is in the meeting room here, Hubert Löwenheim, the head of the ENT clinic, which is what I've heard the second largest ENT clinic in Germany, and he's also heading the renowned hearing research lab in Tübingen. The company has a fairly unique setup. We have a fully virtual setup, which makes internal communication, decision-making a bit easier. But that also means that all the research work, all the clinical work we do first on an exclusive cooperation with the hearing research lab of the university. And then over the last 30-odd years, Jonas and I have developed a good network of experts and high-quality CROs. We have established a fairly strong IP portfolio with 2 granted substance matter patents on the compounds I will cover a little bit later, and we also have 2 pending formulation patents. And basically, the programs we are currently pursuing date back to a, so to say, white smoke meeting, which took place about 7 years ago, were based on literature, this research, and expert advice. And I think that meeting that was before my time took place at the Frankfurt Airport Center. The decision was taken to pursue KV 7.4, the KV 7.4 in the outer hair cells as the target for a therapeutic approach towards treating SNHL. Now, since then, in variance to other companies, we've not repurposed established drugs, but we started with a high-throughput screening of compound libraries, went through the process of lead optimization, which Jonas earlier pointed out, did all the preclinical work, including toxicology and safety testing, and we have in the meantime reached the late preclinical stage with ACO 82, which I will in part also cover in this presentation. And we're in the midst of the clinical phase 2 study with ACO 85 or Bimokerna. Let me just see what yeah. Now, our research and development program complements very nicely the academic and industry-wide efforts to make the global hearing loss epidemic a druggable disease. You all know in the audience most of the details on this slide, so I would like to focus your attention to the lower right box only, which gives the fact that roughly 90% of all etiologies of SNHL is basically driven by the loss of function of outer hair cells, the main sensory cells in the inner ear, and finally the cell death, the apoptosis of these cells. This has been confirmed in human temporal bone histology examinations over the past years. It has been proven for sudden sensory neural hearing loss. It has also been shown for cisplatin-induced ototoxicity, and more recently, also for age-related hearing loss. So the loss of function and the Cell death of outer hair cells is the major contributing factor to most forms of sensory neurohearing loss. And that's why we looked for a target in this white smoke meeting, which I quoted earlier, in the outer hair cells. Kv7.4 channels, as you probably all know, are crucial for hearing. They are potassium efflux channels. They are primarily expressed in the auditory system. And what they do in the outer hair cells, they maintain homeostasis, they repolarize cell membranes for rapid sustained function, which is important since sound is a continuous stimulus. So these cells need to be in a ready state very quickly in order to pick up the next sound. The Kv7.4 channels are biologically validated targets, and they ensure functional hearing with tuning and amplification of the cochlear function. A point mutation or different point mutations on the KCNQ4 gene of the Kv7.4 channel leads to DEFNA2A. And we also know from more recent GWAS studies that the Kv7.4 channels play a very important role in age-related hearing loss. We've tested our compounds in vitro, and they do restore mutant Kv7.4 channel, but also increase wild-type currents by the factor of 2 to 5 with a low nanomolar EC50. And the mode of action is twofold of the compounds. And we've seen that in disease models in vivo, but we've also seen it in healthy animals tested. Initially, we see an acute hearing enhancement through a functional outer hair cell improvement. And then long-term, we see a outer hair cell preservation through control of pathologic depolarization in acute and chronic progressive hearing loss. So the compounds have a dual mode of action. You might also say a bimodal mode of action, and maybe there's the connection for easier memory. So bimocalma has a bimodal mode of action. We have two compounds in development. Focus today is on the advanced phase 2 program with ARCO85, but we also have ARCO82. They have the same target, the Kv7.4 channel, which they activate, but they differ in terms of physicochemical properties. ARCO82 is well absorbed in the intestinal tract, so it's being developed as a tablet formulation for all the chronic forms of SNHL. But we're at an earlier development stage, preclinical. ARCO85 is poorly absorbed and therefore has to be injected transtympanically into the middle ear, into the round window niche, from where it penetrates into the inner ear in order to reach the outer hair cells. So it's a local delivery and therefore better suited or best suited for acute and subacute forms of SNHL. It's an etiology-agnostic autoprotectant, and I'll show you that on the next slide, aiming to minimize permanent hearing loss in acute or subacute forms of sensory neurohearing loss. We have, in the meantime, developed a so-called thermoreversible slow-release formulation. That's a formulation which is liquid at 7 to 8 degrees centigrade, so in the fridge, not in the freezer, and then becomes gel-forming at body temperature. And therefore, the injection through the tympanic membrane into the round window niche leads to a gelification of the formulation, and the formulation stays there. And I'll show you later on PK data, which indicate that there are high intracochlear concentrations over a period of 2 to 3 weeks after a single injection. Now, the local application of bimocalma, of ARCO85, also brings the advantage that we have a very low systemic exposure, which minimizes drug interaction, which also Jonas pointed out earlier, which is of particular importance in oncology indications where you don't want to interfere with the efficacy of chemotherapy. We have, in the meantime, completed a phase 1b study. I'll briefly have a summary on that later on. In a collective of patients with stable symmetric age-related hearing loss, since that's a slightly invasive procedure. We didn't select healthy young volunteers, but looked for elderly patients with a stable form of age-related hearing loss, and we're currently in phase 2, as said earlier. Now, both compounds have been tested in vivo in a variety of animal models. We've looked at otoprotection against aminoglycosides, very effective, very robust, reproducible results. We've looked at otoprotection in cisplatin-induced ototoxicity. Also, on that one, I'll show you the details later on. We've also looked in 2 mice models on the treatment and prevention of presbycusis, age-related hearing loss in mice, and we've also done some studies in healthy animals. And so we have a broad range of potential indications for the compound, which is a nice problem to have. And that brings us to the question which was asked earlier. If you have a compound or a concept which has a broad possibility of applications, which indications do you choose for your clinical phase 2 study? Not an easy one. Do you choose DEFNA2A, like an orphan drug indication, small indication, has certain advantages? Do you look at sudden sensorineural hearing loss, noise-induced hearing loss? That's, by the way, the only preclinical model we've not tested in detail. Presbycusis, aminoglycoside, or cisplatin-induced ototoxicity. So what we had was a second sort of white spot meeting where we looked at the strengths and weaknesses, opportunities and threats of these different indications, looking at clinical feasibility, preclinical feasibility, the technical risk of development, the commercial potential. So a number of factors. I will not mention all of them, but we did a systematic analysis in-house with our experts, with our shareholders, in order to find, from our perspective, for the Kv7.4 activation, the ideal indication. And the decision in the end was, let's go for cisplatin-induced ototoxicity. Now, you're all aware of cisplatin-induced ototoxicity. Cisplatin introduced into the market here in the States by BMS in 1978, the so-called penicillin of oncology, remains a crucial element in cancer treatment. On a worldwide basis, still every year, more than 500,000 patients with different forms of solid cancers, lymphomas, leukemias, receive cisplatin-based chemotherapy regimens annually. And according to more recent publications, about 70 to 80% of adults receiving cisplatin at a dose of at least 300 milligrams per square meter body surface. That seems to be sort of a threshold dose for an increase, a steep increase of the incidence of ototoxicity. 70 to 80% encounter clinically relevant hearing loss. That's the result from more recent publications. If you look into the older publications, the incidence is in the range between 30 to 50%. What is the difference? The newer publications have applied high-frequency and extended high-frequency PTA measurements and thereby also captured the effects of cisplatin on the higher frequencies. And that's the driving difference between the incidences you find between more older publications and newer publications. Now, that hearing loss affects communication, social interactions, emotional well-being. You're all aware of that. But is that a long-term issue for these patients? There's a study called the Platinum Study. It's a longitudinal follow-up study of cisplatin-treated testicular cancer survivors. Actually started off by one of the pioneers of cisplatin treatment, Larry Einhorn from Indiana University. So 2,000 patients with baseline measurements and then follow-on measurements. They were recruited between 2012 and 2018. And from that large cohort, a sub-cohort of 100 patients with a median age of 48 years, and a median time since chemotherapy of 14 years were assessed thoroughly audiometrically. They all had normal baseline PTA levels before cisplatin therapy. And after 14 years, 80% of participants displayed a fairly drastic, audiometrically defined hearing loss at follow-up. So it's a long-term issue for these patients. There's a high medical need. Before moving into that indication clinically, of course, we did preclinical in vivo studies. This is from a guinea pig experiment. These guinea pigs received round window electrodes implanted and then received a local ototoxic insult with cisplatin over 15 minutes. So the initial insult was set, and then thereafter the animals were treated with a local ACO85 in 3 different dosages, or vehicle, placebo, via mini pump, because at that time we didn't have the slow release formulation yet, over a week. And after 2 weeks, compound action potential threshold was determined. And what you can see on the right-hand side in the graph, the dotted black line, that is the placebo line. You see a fairly drastic increase of CAP threshold by 60 to 90 decibel, whereas you see a dose-dependent decrease of that effect via the 1-week infusion of ACO85 in 3 different dosages. So there is a protection of the cisplatin-induced hearing loss in these animals, but is there a histopathological correlate? So how does that look from the histopathology side in the same experiment? So what we did was, or what was done in the lab was a whole mount preparation of the cochlea, and the outer hair cells, the number of outer hair cells per millimeter distance from the cochlear apex were counted. Now, the normal value in this animal model is around 300 outer hair cells per millimeter. And what you can see again, the black curve is the placebo curve. So the animals receiving placebo lost about 2/3 of their outer hair cells, whereas all 3 dosages of ACO85 basically protected the outer hair cells entirely. So in these animals, almost all outer hair cells were still present 2 weeks after the insult. One more slide before I come to the clinic. We have, in the meantime, as said earlier, developed a proprietary sustained release formulation, a thermoreversible formulation. And what you can see here also from a guinea pig experiment, after a single transtympanic injection of that formulation, you see over a period of 2 to 3 weeks, fairly high levels of compound in the inner ear tissue and in the perilymph, whereas you see very limited, if any, systemic levels in the blood plasma. So the duration of action and effective levels at the site of action at the outer hair cells can be achieved over a period of 2 to 3 weeks. Quick summary of the phase 1b study we did in elderly volunteers. We included 15 patients. The initial 3 patients received 1.2 milligrams of ACO85 in a volume of 200 microliters into the round window niche. There were 2 baseline assessments of hearing in order to make sure that these were patients with stable hearing loss, and then there were consecutive examinations, including PK. We saw very mild and transient adverse events, which were mostly procedure-related to the transtympanic injection. And the clinical review board and we then decided to move to a higher dose, 6 milligrams, again in 200 microliters of formulation, 12 patients treated, mild and transient adverse events, procedure-related. We saw even in the high-dose group peak plasma exposures below 2 nanograms per ml, so a negligible plasma or systemic concentration, and the desired long residence time in the middle ear was confirmed in the study. Now, if you move into phase 2, in our case, we didn't have a clear picture of the expected effect size of the drug, if it works. We didn't really know the variability in our target group, so patients with cancer being treated with cisplatin. And if you want to control the power of your study or increase the power of your study to the extent possible, there's two main levers. You can work on variability of your patient cohort, and you can also try to optimize the outcome incidence in order to show with a smaller study a more pronounced effect. And that's what we did. So if you look at individual rather than intergroup comparison, of course, you have a much lower variability. And what we did was we designed, as you can see on the next slide, a so-called split body trial design, which is probably familiar to you, at least from the ophthalmology field. So we designed a study where we, within one patient over time, treat one ear on a randomized, double-blind, placebo-controlled fashion with active principle, and the contralateral rear in a randomized fashion with placebo. We've focused on a single cancer type in order to have a very homogeneous group of patients, and on healthy hearing subjects. So as you can see on the next slide, we are only in this first phase 2 study looking at young male hearing healthy patients with testicular cancer. The third topic also came up, I think, during the discussion here already. Speech understanding testing between different languages leads to variability, to say the least. So we've, in this first study also, since we're based in Germany, focused on German as the only language for the speech understanding, because we are applying a very wide range of hearing tests. And on the outcome incidence, the guideline for the chemotherapy of patients with testicular cancer with a certain staging of disease ask for at least 300 milligrams of cisplatin per square meter body surface. So a high dose or higher dose even than 300. And what we are applying in this study as the main readout is a extended high frequency PTA up to a 16 kilohertz. And we are also measuring DPOAE as a function of the outer hair cells, direct function readout of the function of the outer hair cells up to 10 kilohertz. That's the study, as said, in a group of testicular cancer patients receiving ototoxicity. It's a clinical phase 2 study approved by EMA and BIFA, split body design, which I've explained. Now, therefore, you only see, even though it's a placebo-controlled study, one arm in this study. These patients receive at least 3 cycles of cisplatin-based chemotherapy regimens. Most patients receive a PEP regimen. And we use the time window invariance to the animal data I showed you earlier. We use a time window of 0 to 48 hours before the start of the cisplatin infusion to do the transtympanic injection, so to prime the ears with IMP or placebo on the contralateral ear, and then the cisplatin infusion takes place. And there's a 3 weeks interval until the next infusion. And again, time window 0 to 48 hours before the second infusion, time window 0 to 48 hours before the third infusion. We do the IMP and placebo injections, which matches very nicely the expected duration of action, effective local drug levels over a period of 3 weeks. In terms of hearing tests, I think I've gone through that. We do these measurements at every visit. The main readout is at the end of the third cisplatin cycle, because some of the patients may receive further cycles of cisplatin, and therefore we've chosen that one as the main readout. First visit took place in the first quarter of last year. We have 15 German high-volume study sites participating. Not all of them have been initiated because of administrative hurdles in Germany. We have right now, as we speak, 10 centers active. And the study is expected to be clinically completed by the end of 25. So we're in the middle of the study. As of Monday, we'll probably have roughly 50% of the patients included. Data are fully blinded. What I can say already at this stage is that so far all patients who have completed visit 4 have experienced, according to ASHA criteria, ototoxicity. So very high incidence of ototoxicity. In summary, outer hair cell dysfunction and loss drive sensory neural hearing loss. Our Kv7.4 activators, ARCO82 and Bimocalna, restore DEFNA 2A mutant channel function, but also increase wild-type channel function in vitro. The dual or bimodal mode of action acutely enhances outer hair cell function and increases outer hair cell resilience and survival. ARCO85 reduces hearing loss and outer hair cell apoptosis in a guinea pig cisplatin ototoxicity model. The thermoreversible formulation has passed phase 1, and we're in the midst of phase 2. And due to its etiology agnostic mode of action, we anticipate that ARCO85 will be effective across all types of acute and subacute sensory neural hearing loss. With that, I'm almost at the end. So where are we based? We are based in the midst of old Europe, in Tübingen, a sort of university-driven, university-dominated small town of 90,000 inhabitants, 30,000 are students, hosting one of the oldest universities in Germany, founded in 1477. So a lot of history, a lot of heritage. But we also have probably one of the most modern ENT clinics in Germany on the hill of Tübingen, where we also have our offices. So in case you pass by, you come to Tübingen. It's not so well connected, so you don't come there by chance. But I think there's a good opportunity. In September, we'll be hosting, or the group of Hubert Löwenheim will be hosting the 60th workshop of inner ear biology. So I hope you take the opportunity, come and see us, visit us, and maybe you will already have by then some more detailed results from our phase 2 study with Bimocalna. And with that, I'm at the end of my presentation, 2 minutes ahead of time.
We have time for a question. I have a question. The SWOT analyses that you did as you were looking across indications must have been really challenging and also really interesting. I'm curious about the timing. Had PEDMARK already been approved then, and did that give you a pathway to move forwards more confidently with cisplatin-induced hearing loss?
No, basically at that stage, we had preclinical data available, but this was done prior to starting phase 1. So basically, when we designed the entire development program, including potential further studies beyond phase 2A. So at a very early stage, and we had quite some debate also between different shareholders of the company. Company is a private company, but in the end, I think we had a good conclusion. And everybody's happy with that selection so far.
Thank you.
Hi, thank you for your talk, which was very interesting, by the way. I have a quick question regarding the preclinical data that you showed, especially between the electrophysiology and the histopathology. You had a super nice dose response on the preclinical electrophysiology, but you had some differences on the side of histopathology. How do you explain those differences of results between histopathology and electrophysiology?
Very good point. Here's the I've been asked to take that one. So it all comes back to the very ingenious selection of the INN the bimodal for the bimodal action. So in the dose response testing, you will be seeing a combined effect of the hearing protection and the hearing enhancement, whereas in the histopathology, you will only be seeing the effect of the hearing protection. So basically, you can assume that even there is roughly the same number of hair cells preserved at the time that you're doing, at the same time point, the hearing assessment and the histopathology, due to the long exposure of the drug and especially the long exposure in the tissue that releases back into the perilymph. You would have this boosting effect at the time of you doing the hearing measurements. So the higher doses, the higher concentrations would be delivering more of the boosting effect at that time point than the lower doses.
Thank you.
Brings us to our final presentation for today, which is by Dr. Jonathan Keel from Sound Pharmaceuticals, who is the Chief Executive Officer and Chief Medical Officer. The title of his presentation is Development of SPI 1005 for the Treatment of Hearing Loss and Tinnitus. Please help me welcome Jonathan.
All right, so hopefully I won't go over time. There's drinking afterwards, right? The benefit of sponsorship. Okay. So thank you for this invitation, and thank you for all the wonderful talks that preceded mine. This is just a summary slide. I mean, we all know the MOAs that have been tested in both hearing loss and tinnitus randomized controlled trials. I think it's fair to say, especially with the data that I'll present, we have the only successful molecule that has passed multiple phase 2s and now a pivotal phase 3. The chemistry of Epsilon is unique. It's selenium-based. We have two modes of action that we think are beneficial to the cochlea. One is its ability to mimic glutathione peroxidase 1, which is also a selenoprotein. Two is its ability to induce GPX1 under redox stress, and we know that's a NRF2 dependent mechanism. Okay. So this is some histopathology from a rat study we did years ago. Immunofluorescence in green is where GPX1 is highly expressed. So you see an A before noise. GPX1 is bombing in the stria vascularis, organ of Cordy's, spiral ganglion neurons. After intense noise that causes both a loss of outer hair cells and a significant PTS, you can see the strial edema, Reisner's membrane ruptured. And then in C, again, we started dosing these animals one day before. The stria is preserved. Reisner's is bulging, but it's not ruptured. Organ of Cordy is high, as are the spiral ganglion neurons. We could quantify the level in that graph in the stria vascularis, so it goes above baseline. We have 5 active INDs in neurotology indications involving hearing loss and tinnitus. I'll speak about Meniere's disease, which is fast-tracked, our initial acute noise study that was done with Colleen Laprelle when she was at the University of Florida, and our aminoglycoside ototoxicity that was done with the help of the Cystic Fibrosis Foundation. This is from that Lancet paper that was published in 2017. We were a little frustrated trying to work with the Department of Defense and test our drug before, during, and after a noise exposure. A lot of controls are necessary to determine the TTS, and then a defined noise exposure, and then a quiet period to see the PTS of all. And we tried for 4 years, double CRADA, double IRB. I went out to certain sites, you know. And, well, thankfully, Colleen was dialing in what we affectionately call the calibrated sound challenge, the iPod player protocol. And it satisfied a lot of the FDA requirements. How will you control for the source of sound? The intensity, duration, use or non-use of hearing protective devices. So this was run by audiologists, a very intensive protocol, 2 dB steps. We did 8 audiograms in a two-week period. Oh my God. The day of exposure was a very long day. So young adults with no hearing loss, no tinnitus were randomized to one of 4 different groups, so placebo, a low, medium, and high dose of Epsilon. It was only 4 days of dosing. So we started 2 days before the calibrated sound challenge, the day of, the day after, and then followed the patients for 1 week up to 1 month. So what we were looking for is a reduction in the threshold shift. Because Colleen had dialed in the peak noise notch, a very slight one at 4 kilohertz, we feel we could power this interventional study with as few as 20 subjects per arm. And there were a lot of critics of that, thinking this is underpowered. But we did hit our primary endpoint, and we saw a greater than 50% reduction in the severity of the noise notch at 4 kilohertz. That was achieved with the 400 milligram dose group. We did some secondary endpoints that combined 3, 4, and 6, 4, 6, and 8. Our biostatistician wanted all frequencies, like an area under the curve. And then in looking at the distribution and variance of the data, we noticed some ears were big shifters, as we call them. There was a high asymmetry across the cohort. So when we did that responder analysis, a 10 dB shift, it's not advancing now.
You're right. See. Nope, we're totally frozen.
It's frozen. Okay.
Good job, Jonathan.
Well, you know, I get to buy a little more time at the end. It only costs another glass of wine for everyone. Is there a limit in March?
The limit is 1 hour.
Oh,
There we go.
excellent. Very good.
Okay, thank you.
So if we use that 10 dB shift from baseline as a responder criteria, and if you have one or both ears, that's a response, the proportion over the next 4 time points post-calibrated sound challenge is high in the control group. So in the control group, it could be as high as 60%, and within 3 hours, down to 30%. And all the active groups were less. And some of those differences were significant or bordered on high significance. So that's just a brief summary of what we've done in acute noise. We've done some, I think, substantial work in ototoxicity, both with cisplatin, aminoglycosides, including tobramycin and amikacin. I'm going to focus on our aminoglycoside work. This is work done in mice, both in cochlear cultures and in live animals, followed for months after an exposure to tobramycin for 2 weeks, and epsilon dosing at 1/10 that dose can be otoprotective. We've tried to translate this work into clinics, so we scored ears using a modified ASHA criteria, and again, we see this temporal peak in ototoxicity at 8 kilohertz in mice, not at the highest frequencies tested. And this is something we've also seen in our human CF study. While we can test out to 20 kilohertz, they don't necessarily have that peak shift at 20, 18, sometimes even 16. We see it typically at between 12 and 14 kilohertz. We've done the same with amikacin. Amikacin is very interesting in that we need a higher dose to get the type of ototoxicity we saw with IV tobramycin. But again, epsilon held at 20 mg per kg is still very otoprotective. What was very interesting, and this is work done by Ryan Longenecker when he was in our lab and brought in gap detection, a modification of pre-pulse inhibition, was that the majority of animals all developed hearing loss. A subset developed these gap detection deficits, and then a smaller subset developed what we think is hyperacusis, meaning their startle response over time went above baseline. Normally, these animals habituate over time. And it was remarkable. It was significant. And then just to summarize our phase 1/2 in cystic fibrosis patients with acute lung infections now requiring IV tobramycin. Again, this was co-funded by the CF Foundation. This first publication is from Emily Harris' work. It was published in JCF. And what we see here, this summary table, using ASHA criteria, is the proportion of subjects that developed hearing loss in the conventional frequencies, in the upper octave of hearing, the sensitive range for ototoxicity using the NCRAR protocols, and the combination. So 2 weeks after that 2-week course of IV tobramycin, 89% showed ototoxic change. It went up to 93% a month later. So the 2 follow-ups were 2 and 4 weeks after they had stopped the IV tobramycin. We looked at changes in the dp gram, 5 dB, 5 dB times 2, 5 dB times 3. We tested within 4 to 8 kilohertz based on Gada, Malki, and Sally Dawson's work from UCL. And we found it to be less sensitive than pure tone audiometry, and I think others have shown that, like Kelly Rivas. We looked at the words and noise test and the change that occurred, and 40% of patients showed a little decrease in their speech descrims. TFI and vertigo symptom scale didn't change that much. But what's equally interesting on the right-hand table are the preponderance of unilateral. So it was highly asymmetric. In fact, in the conventional audiogram at 4 weeks, it was all unilateral. DPOAEs, again, sensitive, but not as sensitive as pure tone audiometry. And here's a graphic from our interventional study. We had randomized about 40 subjects to those 4 different dose arms. Again, 15 days of IV tobramycin. We measured at 2 and 4 weeks. We scored them using ASHA criteria. In 2 weeks, we see the placebo rate is 71%, and then at 4 weeks, it goes to 100%. So we do see this increase. The active rates started out as high as 100% or 75%, but they went down to 86, 44, and 43%. And it was significant with the 400 mg dose group. The 600 mg dose group actually had 2 fewer people, so it was not statistically significant, although it bordered on that. And here's just a summary. Again, the 400 mg group was 0.03, the 600 mg group was 0.07, and we have 2 more studies that we plan to do. Just briefly, I'll mention we're collaborating with MED-EL in a future cochlear implant study where we'll use our drug SPI 1005 starting 2 days before implantation and continuing for 6 months. And then we'll follow the patients for a total of 12 months to help preserve low-frequency hearing post-implantation. We are presenting a poster on the animal work that provides the proof of concept. So while I put them here, I thought I'd just click through. It's something we can discuss in the poster session. So we can now focus on our phase 3 result in Meniere's disease. So this is the largest Meniere's study completed. We randomized over 220 patients. We treated them for 1 month, followed them for 3 months, and then put them on to open label extension for up to 12 months. Meniere's disease was reclassified in 2015. There used to be 4 classifications, now there are 2. And we bolded some of the most significant changes that happened in the 2015 reclassification or amendment to the diagnostic criteria. 1, they put an upper limit on vertigo of 12 hours. 2, audiometric documentation of low-frequency hearing is required now. And for probable, they now allow for dizziness, and again put an upper limit. So what used to be considered a disease of vertigo has shifted to an auditory vestibular disease, where for the definite diagnosis, you need low-frequency hearing loss in the affected ear. And in recent years, there have been several systematic and meta-analyses of the published literature involving clinical trials with Meniere's patients. And whether it's Basura 2020, the updated guidelines for clinical practice in Meniere's disease, Webster 2023, the Cochrane database reviews, or Taniguchi, a review of control groups in Meniere's trials, essentially the conclusion is very similar. There is low to very low evidence of efficacy, even for corticosteroids. I think we knew this about the antivirals, diuretics, beta-histine. Beta-histine is not approved in the US, but is approved in Asia and Europe for dizzy patients. So on to our pivotal phase 3, the STOP-MD trial. We looked to enroll at least 200 patients up to 75 years of age. Again, low frequency hearing loss, active symptoms. The FDA wanted us to enroll patients with tinnitus, oral fullness, vertigo, or dizziness. We allowed them to continue on their standard of care except for concomitant steroids. We were concerned it could mess with our safety or efficacy signal. So the RCT, again, 1 month of treatment, 3 months of follow-up. Every month we reassess them from baseline. Open label extension for up to 12 months, and it was only done for safety. Epsilon is a new chemical entity. It's never been approved anywhere. So we have to do a certain number of patients for safety, but every 3 months we brought them in and reassessed them. We used pure tone audiometry, the words and noise test. Because this is a pivotal, we had to have 2 co-primary endpoints, a 10 dB improvement in low frequency PTA, a forward improvement in the WIN score, and our baseline patient comes in scoring only 15 of 35 words correct. We use these PROs that I'd mentioned in a Q&A before, the TFI. We look specifically at tinnitus loudness, which is question 2, the vertigo symptom scale. We set up the oral fullness scale, which is analogous to the tinnitus loudness scale, and then the dizziness handicap inventory. We added the DHI in because a lot of our older patients no longer complain of severe vertigo. Many of them say, I haven't had vertigo in a decade. Occasionally I get a little disequilibrium, but it's brief. So we thought that may be more sensitive. So the primary objectives, safety, 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 co-primary endpoints based on adopted responder analyses, and then these different secondary objectives. Open label extension, again, every 3 months we brought them back into clinic, did the full reassessment. So we're still mining that data, but it looks very supportive for chronic treatment. So again, this is the 1, 2, 3, 4 of the RCT. This is what the consort flow diagram looks like. Again, 254 screened, 221 randomized, 106 completed in each group. This is what's called the intent to treat. So once randomized, always analyzed, they took at least 1 dose of drug. This is their demographic, mid to late 50s, slight predominance, male, mostly white. Now onto per protocol, we had to eliminate 29 subjects. Not everyone was compliant during the RCT, and at least a dozen patients took a concomitant steroid. So this is their demographic, very similar. Again, slight preponderance male, predominantly white. All right, so all the data after, at least during this RCT period, are per protocol. So the FDA required these primaries, a co-primary, to be analyzed independently using Fisher's exact test. They wanted the critical difference to be at day 84. So we achieved that. In 2 of the 3 follow-up time points, active outdid placebo in a robust way. This is some additional analysis that was pre-specified. We looked at the dB change at 250, 500, and 1,000, and an average of 250, 500, and 1,000. And you can see in gray bars the placebo group, they all got worse at day 28, day 56, and day 84. And the active groups, well, the active group got better. And this difference is about 5 dB. So with 1 month of treatment and 3 months of follow-ups, we're seeing significant differences in their audiograms. This is the words and noise test score. Again, the improvements from baseline. Placebo, on average, is plus or minus 0.3 words. Baseline, again, was 15 words out of 35. So it's approaching almost a 2-word improvement in the active group. Now, if we look at some of our secondary endpoints, the FDA actually wanted us to show a 10 dB improvement at 2 adjacent low frequencies. We moved that to the secondary endpoint. I'm a little scared we couldn't achieve that, but actually the data look even better from a relative scale. It's kind of a little stairway to heaven there. Placebo is 11 to 14%. Active rate continues to go up through day 84. A 20% improvement in the WIN score was non-significant, and we think it's too sensitive. Again, a 15 becoming an 18 is technically a 20% improvement. That would only be a 3-word improvement. And then here are the improvements in the different PROs. No significant differences here during the RCT for tinnitus functional index, tinnitus loudness. What was interesting, though, if we did a responder analysis looking at the TFI, an 18-point improvement. Now, our patients typically score 51 points at baseline, so they're highly affected. Within the TFI, the greatest treatment effect using Cohen's is about 1.5 with an 18-point reduction at 6 months. Using that responder criteria, we had a significant difference at day 84. The data look pretty good. What I like about this responder analysis is that we drove the placebo rate flat, and the active rates are better at each time point. It's almost a doubling at day 84. And then here are the other PROs, again, non-significant differences or improvements during the RCT follow-up. And then on to open label extension. Here we had 153 complete 6 months and 82 complete 12 months. And if we use that same 10 dB improvement at one of the low frequencies, the responder rates now go up. So essentially what we did is bring the placebos up. The placebo rate before was only in the 30s. The active rate was in the 50s. Now, if you continue on this drug, a little bit more than 50% showed this 10 dB improvement from baseline. And these patients have been on study now for sometimes 15 months. Meniere's patients that are in their late 50s typically decline. We saw a nice improvement in the WIN score. Again, we're bringing the placebos up, so this is higher than the average rate from the RCT. And then here's the rates if we use those more strict responder criteria, the 2 times 10 dB improvement. And now if we look at the PROs, their reductions from baseline, they're all significant. During the RCT, they weren't. But now being on the drug for a longer period of time, the entire cohort is averaging about a 15-point reduction in TFI. Again, they start off at 51. Tinnitus loudness is now improved from a 6.6 to about a 1.5 point reduction. Vertigo symptom scale, oral fullness scale, dizziness handicap inventory. Nice significant reductions in their scores. And so here's a summary. This drug was pretty well tolerated. We saw, a series of mild and moderate adverse events. We had 2 SAEs during the RCT. One person had a substantial loss of hearing which actually we expected since we enrolled people with active Meniere's disease. And we had 5 SAEs during the open label extension for 6 months. Again, none of these AEs were thought to be related to study drug. So we hit our co-primaries. We wanted to show at day 84 a 50% improvement over placebo, so we were able to do that. Open label extension looks very supportive, not just from a safety perspective, but for a continued improvement perspective. And this summary table is of the 13 studies we've completed, enrolling about 800 patients. The 4 studies in yellow are studies that we're looking to enroll this year or partially enroll this year. It'll be harder with the ototoxicity studies since that's a rare disease, at least in the CF population. So I think that's it. And I just wanted to thank you for your attendance today.
All right, we will move to open discussion. Questions from the audience related to any of the presentations that we've seen today? Jonathan, I have a question for you. Particularly to help some of the basic scientists in the room who are very used to looking at animal data just from a perspective of, is there a statistically significant difference? And you introduced the idea of differences that reach a clinically important criterion. So could you talk a little bit about minimum clinically important differences and some of those conversations with the FDA? I was struck by your phrase, the FDA is looking for, and so it may be helpful to people to hear a little bit about what that looks like.
Yeah, I think from our experience, at least with ototoxicity, the ASHA criteria are well accepted. My sense is that unlike what Fennec was able to achieve in peds, adults will require not only an improvement in the audiogram or reductions in cochlear toxicity, but an improvement in speech discrimination or some other measure. I think the FDA is going to push everyone to a co-primary. That's my sense. So hanging your hat on the audiogram, otoacoustic emissions, I don't think so. Yeah.
Tim, do you have anything you'd like to add about the clinically important testing and your interactions with regulatory agencies about selecting endpoints?
But so far we've been only in interaction with the BfArM, so the German authorities, at the outset of our program, and there was no detailed discussion on outcome measures. So what we're doing in our study here and in further studies to be done is we look at also at speech understanding and noise and quiet. So we do in our case, the Freiburger and the Oldenburger test. But I think in that sense, the project which was presented by Ralph is very important, since my understanding is so far there is no coherent agreement what the relevant readouts in the end need to be and what the effect size needs to be. So this is, I think, still open, and thereby the cooperation between industry, academia, and regulatory affairs is going to be very important to give more certainty whether your trial program actually is going to meet the requirements or not.
Did I see a hand? No, okay. I guess this plays really nicely into some of your goals as well, Ralph, with looking at clinical measures and what to recommend and what to standardize. Do you think that when you start assembling a group, they'll be tasked in particular with looking at what are the minimum differences necessary, going beyond what measures to look at?
So I think I heard your question properly. So I think, yeah. So I think the key thing is the meaningfulness. So what's meaningful to a patient and the clinician. And I guess the trick is going to be working out how do you measure that meaningfulness.
Yeah, I agree. Oh.
The drug for Meniere's treatment is quite promising because Meniere episodes could be with gap, couple of days, couple of weeks, or even months. So when the treatment's actually provided, at what stage of the Meniere's disease, during the episodes or between the episodes?
Now, the majority of our data were all accrued from active patients, but we do have some patients that were 28 years into their disease. So 2/3 of our patients are unilateral, but 1/3 are bilateral. They're highly asymmetric. So typically the bilateral patients were older. They'd suffered for at least 10 to 15 years. Their second side became active. So that's why they enrolled in the trial. I think, rarely enrolled a naive patient. All of them were on a standard of care, had been to see their PCPs, an ENT, a neurotologist. In fact, all but one of our investigators has been a neurotologist. So these are patients that are suffering. What's interesting to note is that as the patients age, certainly beyond 5 years, severe vertigo may not be their primary complaint. They do have vertigo, but it may not be of that 20 minutes duration anymore. Oftentimes they'll have more frequent disequilibrium, but the progressive hearing loss, the fluctuating hearing loss, the oral fullness, and tinnitus become significant. So Michael Ruckenstein did a really nice publication recently where they looked back at the UPenn experience over 9 years, pulled out all their Meniere's patients by ICD-10 code, and then reclassified them using the 2015 amended criteria. And the numbers went down from 806 to 480 to 168. Only 168 of the 806 had definite Meniere's, by their EMR at least. Their leading complaints were all auditory: tinnitus, fluctuating hearing loss, oral fullness. So it's a complicated disease. You know, there are debates about why patients burn out of their severe vertigo within 5 years, and why dizziness becomes the more dominant vestibular feature. And then, of course, you know, a small proportion of patients can go to deafness. They've received cochlear implants. So we've taken kind of all comers, and the ease of our drug being administered orally, I think, helps with the adoption.
Thank you.
I have a question for all of the members of the panel. We have a number of groups here who offer a lot of different animal models, preclinical models for testing, and also people who are doing clinical trials who are reliant on animal models. I'm curious what you think the models are that we're missing. What are the gaps where you have a hard time providing models for people who are working in a particular target disease indication? What are the diseases you hope to one day address where you lack animal models?
So I think the most missing model is an age-related hearing loss model, because there is clearly no standard for that, and this is really missing. We have worked previously with the SAMP8 mice, but this is not anymore an age-related model. We have performed different studies, and we are not able to see this progressive hearing loss anymore. So, and we have a grant for that this year, that we will develop a new model for this age-related hearing loss, but with no guarantee that it will work.
Yeah, so I certainly second that. And I think that certainly animal models for more complex disorders and the central auditory system will become even more important. So we know disorders such as tinnitus often originate in the cochlea, but as the disorder progresses, there are more central auditory structures, attentional and emotional networks that are affected. And so I think having better models and a better understanding of those disorders and the variations of those disorders would be really important for therapeutic development.
Just to add on to that, one of the things we were talking about sound for the last 5 years is age-related hearing loss. And, you know, with one of our scientists, I was saying, okay, we're going to need to age up mice for at least 6 months, then maybe repeatedly expose them to noise over 6 months, and then watch them age another 6 months, and do an intervention at 6 months, 12 months, you know, and male, female. This would be a massive undertaking and probably require about 600 mice.
I'll just add that you can actually just take standard Wistar rats and let them age. And we happen to have a poster jointly with the group at Hoover Neuenheim on that tomorrow. So I suggest you go meet Matthew Peterman and see how you can monitor this over time.
Yeah, question to Jonathan. Thank you very much for the great data set. So if we're taking this right, you included patients at different stages of their Meniere's progression, which included patients which had like a few years of progression already. And a little speculative question, if you were to catch patients in the very early phase of the disease where, you know, hearing fluctuates back to normal, do you think if you start patients at that stage, you could actually prevent the progression of the disease or influence that?
Yes, thankfully we have Emily Hariff working at Sound, and we have a post hoc analysis where we think we're disease modifying. And in our phase 1b of only 40 subjects, where we randomized 40 to 4 different groups, we had an N of 10, I was fearful of treating the older patient. So we required patients to be within a year of diagnoses. We actually had a much greater difference between the placebo rate and the active rate. But the FDA wanted us to enroll active patients. They stress this. Yeah.
Hi. Firstly, a comment. So I first attended ARO in 2013, and the landscape was pretty bare then for therapeutics moving forward. It was very much still reformulations of steroids, and there wasn't much genuine drug discovery going on in my mind. But I think, you know, today we've seen a number of talks, and the progression in the field has been fantastic, and congratulate all the teams that have been involved, and there's also the organizations that provide all the services to enable drug discovery. So it's brilliant that the whole field has moved forward. Now, a question I had was, I was struck with, Jonathan, with the data you presented, that once you moved into an open label study, that the self-reporting studies all of a sudden seemed or appeared to be much more positive, whereas in the controlled trial situations, it didn't look like those self-reporting measures were very effective as identifying effects. And I know that coming from pharma, you know, self-reporting endpoints make us very nervous. So I just wonder if, again, you know, wherever possible, we need to find objective measures in clinical studies, but it'd be interesting to get your thoughts on this.
No, I agree with you. I mean, the FDA does like benefit that the patients report, but the Taniguchi paper that came out of MUSC was the first of its kind to do a systematic review of all published RCTs in English. And the placebo rates for vertigo, for example, were 56%. So if you're going after vertigo as your primary endpoint, that's patient-reported. I mean, it's rare for us to see a patient in the clinic with active vertigo. They had it the night before, the week before. That's tough. The same is true of tinnitus, dizziness. That's why we like the audiogram. It's administered by a clinician in a controlled setting, a booth, calibrated audiometer. Same with speech discrimination. And the fact that the Academy moved to adopting what the Bharani Society and others stated is that we need to document low-frequency hearing loss. That, quite frankly, benefits us because nothing's been shown to improve. With this robust data set, I haven't seen anything. Everything failed. Every antioxidant study failed. Yeah.
I think definitely you need to have some sort of quantitative objective measures to support this, and there are options also outside of audiometry, but they're not well enough developed or well enough accepted yet. For vertigo or instability, you can use accelerometers. That's been looked at both preclinically and clinically. Even in free walking conditions for patients, take a defined walk and look at differences in movement with head-mounted and chest-mounted accelerometers. This could be a fairly objective measure. Might also be helpful that it's not in the middle of a vertigo crisis, but simply looking at the long-term consequences of the vestibular damage. On the tinnitus side, there's a number of efforts ongoing to look at more central measures using various efforts in the direction of EEG or other similar modalities. I think potentially ASSR combined with EEG could be interesting to look at sort of entrainment or oscillatory measures as opposed to hyperactivity measures. But these are things that would ideally be developed to go along with questionnaires or self-reported measures of various kinds. The hearing loss, as Jonathan says, is definitely ahead of the game among the sort of three main components of the inner ear disease field. But I think there's headway to be made.
Yeah, I think one of the equally disappointing things about our field, when I first learned about minimum masking level, I thought, wow, here's an objective measure that you could test in certain tinnitus patients, mask their level, and hopefully it decreases. And then when you follow these patients, it didn't correlate to the clinical instrument data. And the same is probably true going the other way, where you have another objective measure, maybe even endolymphatic hydrops, volume, but it doesn't necessarily correlate with the clinical phenotype. And as soon as that happens, then the FDA will reject it. Yeah. I was actually surprised in our interactions with drug oncology products. We did not propose otoacoustic emissions, but their audiologists requested it. At least at that time, I think there was still hope that it could be as sensitive a measure as pure tone audiometry. And we were all being told that, ASHA and maybe AAA were going to update the guidance for ototoxicity. And that hasn't happened. So I think it's a valuable measure, but they understand it's not a hearing test. So using it as an objective measure, again, you're going to get into that co-primary type thing. Okay, now show us with the audiogram or speech discrimination.
Thank you for the great discussion. Let me ask everyone to give all of our speakers another round of applause, thanking them for their time today. And I want to thank